At this very moment, while you rest and your thoughts begin to drift, an invisible war is being waged within you. Billions of cellular soldiers patrol your bloodstream, lymphatic highways, and tissue borders, distinguishing friend from foe with a precision that would make any security system seem primitive by comparison. This is the immune system, a guardian that never sleeps, never takes leave, and rarely asks for recognition. When it works well, you notice nothing at all. When it falters, everything changes. Understanding this protector, this quiet defender, is in many ways an investment in comprehending your own existence, your resilience, and your vulnerability in a world teeming with microscopic threats. Immunity, at its most fundamental, is the capacity to resist disease. It is the biological equivalent of sovereignty, the ability to maintain bodily integrity despite constant exposure to entities that would compromise it. This sounds straightforward enough until you consider the staggering complexity of the task. Every breath you take carries thousands of particles, some harmless, others potentially lethal. Every meal introduces foreign proteins into your digestive tract. Your skin hosts trillions of bacteria, most of them benign or even beneficial. The immune system must somehow distinguish between the sandwich you ate for lunch, the helpful bacteria fermenting in your gut, your own cells going about their business, and the influenza virus attempting to hijack your cellular machinery. This feat of recognition, this ability to distinguish self from non-self, represents one of biology's most sophisticated achievements. It requires a surveillance system capable of screening trillions of molecules daily, memory banks that can recall exposures from decades past, and strike forces that can eliminate threats while minimizing collateral damage to healthy tissue. Get the balance wrong in one direction, and infections overwhelm the body. Or in the other direction, and the immune system turns on itself, attacking the very tissues it evolved to protect. The evolutionary origins of immunity stretch back billions of years, long before humans, before vertebrates, before anything we might recognize as complex life. Even bacteria possess rudimentary immune systems, molecular scissors called CRISPR that can recognize and destroy viral genetic material. Single-celled organisms have mechanisms to distinguish their own membranes from those of potential predators or parasites. As organisms grew more complex, so too did their defenses. The appearance of dedicated immune cells, specialized organs, and adaptive responses that could learn and remember marked major transitions in the history of life on Earth. In vertebrates, including humans, immunity operates on two levels, often described as two overlapping systems, though this division is more pedagogical convenience than biological reality. The innate immune system represents the ancient, evolutionarily conserved arm of defense. It responds rapidly, often within minutes of detecting a threat, but it does so in a relatively nonspecific manner. Innate immunity recognizes broad categories of pathogens, molecular patterns that betray something as foreign or dangerous. A bacterium's cell wall components, viral genetic material, fungal cell membranes, all trigger innate responses through pattern recognition receptors that evolution has refined over hundreds of millions of years. The adaptive immune system, by contrast, is more recent in evolutionary terms and far more specific. It can recognize individual molecular features, distinguishing not just between bacteria and viruses, but between different strains of the same virus, even between healthy cells and those transformed by cancer. This specificity comes at a cost: time. Where innate immunity responds in minutes to hours, Adaptive immunity requires days to fully mobilize during a first encounter with a pathogen. But it offers something innate immunity cannot: immunological memory. Once the adaptive system has encountered a threat, it remembers, mounting faster and stronger responses upon re exposure, sometimes maintaining this vigilance for a lifetime. These two arms of immunity don't operate in isolation, despite how textbooks might compartmentalize them. They communicate constantly, with innate immune cells presenting information to adaptive immune cells, and adaptive responses recruiting innate effectors to do the actual work of pathogen destruction. The system is better understood as a spectrum of responses, from immediate and nonspecific to delayed and exquisitely targeted, all working in concert toward the same goal: preserving the integrity of the organism. What makes the immune system unusual, almost unique among bodily systems, is its distributed nature. There is no singular immune organ comparable to the heart or brain. Instead, immune function emerges from the coordinated activity of multiple organs, billions of circulating cells, and the lymphatic vessels that connect them. Immune cells are born in the bone marrow, educated in the thymus or other lymphoid tissues, stationed in lymph nodes and spleen, and patrol throughout every tissue in the body. An immune response to a splinter in your finger involves cells communicating across your entire body, signals traveling from the wound site to the nearest lymph node, and reinforcements arriving via the bloodstream. This distribution creates both resilience and complexity. Immune function cannot be localized to one place, and thus cannot be completely disabled by a single injury or defect. But it also means that immune responses must be carefully coordinated across vast distances, at least from a cellular perspective. The chemical language of immunity, the cytokines and chemokines that cells use to communicate, must convey precise information about threat type, location, and severity. Get the signals wrong, and the response can be worse than the initial problem. The challenge of self versus non-self recognition becomes even more nuanced when you consider the microbiome, the trillions of bacteria, fungi, and viruses that inhabit your skin, digestive tract, and mucous membranes. These organisms are genetically distinct from you, clearly non-self, yet the immune system tolerates them, even depends on them. Beneficial bacteria outcompete potential pathogens for space and nutrients, produce vitamins, train the developing immune system, and communicate with immune cells to maintain appropriate activation states. The immune system has evolved not to eliminate all foreign organisms, but to distinguish between commensal, beneficial microbes and pathogenic ones, a far more sophisticated task. Even the category of self is not as straightforward as it might seem. Cells die and must be cleared away, sometimes billions per day in an adult human. Dead or dying cells can be dangerous if not promptly removed, their contents potentially triggering inflammation. The immune system must recognize and clear these cells efficiently without mounting the kind of destructive response reserved for pathogens. Cancer cells, despite being derived from the body's own tissues, accumulate mutations that alter their surface proteins, sometimes enough for the immune system to recognize them as abnormal and destroy them. This immune surveillance against cancer operates constantly in the background, likely eliminating countless potential tumors before they can establish themselves. The immune system must also contend with tissues that are immunologically privileged, sites where inflammatory immune responses would cause more harm than good. The brain, eyes, testes, and placenta during pregnancy all maintain specialized environments with limited immune access, not because these tissues are unimportant, quite the opposite. These are sites where inflammation and the collateral damage that comes with robust immune responses would be catastrophic. The brain cannot regenerate neurons easily, and inflammation in the confined space of the skull can be fatal. The eye's optical clarity would be compromised by the cloudiness inflammation brings. The developing fetus is genetically distinct from the mother, expressing paternal antigens, yet must be tolerated rather than rejected as foreign tissue. These privileged sites aren't completely defenseless. They possess their own immune adaptations, resident immune cells that can respond to threats in more measured ways, anatomical barriers that limit pathogen access, and molecular signals that actively suppress inflammatory responses. But they represent compromises, areas where the immune system's usual aggressive approach must be tempered by other priorities. All of this occurs largely outside conscious awareness. You don't feel your neutrophils patrolling your bloodstream or your T cells surveying for abnormal cells. You become aware of immune function primarily when something goes wrong, when the barriers are breached, and inflammation announces itself with pain, swelling, and fever, or when the system itself malfunctions, turning against self in autoimmunity, overreacting to harmless substances in allergy, or failing to respond adequately in immunodeficiency. For most of your life, immunity is silent, effective, and invisible. Understanding immunity requires accepting a certain level of complexity. There are no simple explanations that don't sacrifice accuracy. The system involves dozens of cell types, hundreds of signaling molecules, genetic mechanisms that deliberately introduce mutations to generate diversity, and regulatory networks that would challenge any engineer. Yet certain principles emerge from the complexity. Redundancy is built in at every level, ensuring that no single failure completely disables defense. Specificity is balanced against speed, with rapid but crude responses buying time for precise but slower adaptive immunity. And throughout, the system walks a tightrope between two dangers: insufficient response leading to infection or cancer, and excessive response leading to autoimmunity or chronic inflammation. The landscape of immunity is vast, and we are only beginning our exploration. To understand how this system defends you requires examining its architecture, the organs and vessels that make up its infrastructure. From there, we will meet the cellular defenders themselves, the white blood cells that perform the actual work of immunity. We will explore how they communicate, how they recognize threats, and how they eliminate those threats while sparing healthy tissue. We will trace the education of immune cells, the selection processes that teach them to distinguish self from non-self. And we will confront the system's failures, the ways that immunity can harm as well as protect. But first, we must map the physical territory, the lymphoid organs and tissues, where immune responses are initiated, coordinated, and executed. These structures form the foundation upon which all immune function rests. If the immune system were a nation, the lymphoid organs would be its cities, command centers where strategies are planned, forces are mustered, and responses are coordinated. These structures are scattered throughout the body, connected by an extensive network of lymphatic vessels that parallel the cardiovascular system. Unlike the circulatory system, with its high-pressure pump and closed loop of vessels, the lymphatic system operates at low pressure, moving fluid in only one direction from the tissues back toward the heart. This fluid, called lymph, carries not just excess interstitial fluid, but also antigens, debris, and immune cells traveling from one location to another. The organs of the immune system divide into two functional categories: primary and secondary lymphoid organs. Primary organs are where immune cells are born and undergo their initial development and education. Secondary organs are where mature immune cells take up residence, encounter antigens, and mount immune responses. This division reflects the system's need to both generate diversity in immune cell receptors and then select for cells that will be useful rather than harmful. The bone marrow serves as the birthplace of all blood cells, including the immune cells known as leukocytes or white blood cells. Nestled within the hollow spaces of bones, particularly the pelvis, vertebrae, ribs, and the ends of long bones like the femur, the bone marrow is a soft, gelatinous tissue rich in blood vessels and stem cells. In infants and young children, most bones contain red marrow, actively producing blood cells. As we age, much of this red marrow converts to yellow marrow, composed primarily of fat cells, with active blood cell production increasingly concentrated in the flat bones and vertebrae. Within the bone marrow, hematopoietic stem cells possess the remarkable capacity to differentiate into any type of blood cell. These stem cells divide asymmetrically, one daughter cell remaining a stem cell to maintain the population, the other differentiating into increasingly specialized progenitors. The myeloid lineage gives rise to red blood cells, platelets, and several types of white blood cells, including neutrophils, eosinophils, basophils, and monocytes. The lymphoid lineage produces the lymphocytes, B cells, T cells, and natural killer cells. This continuous production is necessary because many immune cells have short lifespans, some surviving only days before they must be replaced. B lymphocytes, one of the key cell types in adaptive immunity, complete their entire development in the bone marrow. The B originally stood for bursa of Fabricius, an organ in birds where these cells were first identified, but in mammals, the bone marrow serves this function, making the naming somewhat convenient, if historically inaccurate. B cells undergo a complex maturation process in the marrow, learning to produce antibodies and being tested for their ability to avoid reacting against self-antigens. Those that pass these tests are released into circulation as naive B cells, ready to encounter their specific antigen but not yet activated. T lymphocytes, by contrast, are only born in the bone marrow. They leave as immature cells and migrate to a different primary lymphoid organ for their education, the thymus. This small, somewhat mysterious organ sits just behind the sternum in the upper chest, above the heart. It is largest in children, sometimes weighing up to 40 grams, and gradually shrinks with age in a process called involution. By adulthood, much of the thymus has been replaced by fat tissue, though it retains some function throughout life, just at a reduced capacity. The thymus is where T cells undergo one of the most selective processes in the body. Only about 2% of T cells entering the thymus will successfully complete their education and be allowed to leave. The rest will be destroyed, failing quality control checks that ensure T cells can perform their function without attacking the body's own tissues. This process, called thymic selection, occurs in two stages. Positive selection ensures that T cells can recognize the body's own MHC molecules, the cellular identification cards that display samples of internal proteins. Negative selection eliminates T cells that react too strongly to self-antigens, preventing autoimmunity. The thymus is organized into an outer cortex and inner medulla. Immature T cells, called thymocytes, enter the cortex and interact with epithelial cells that present various antigens. Those that can bind to MHC molecules with appropriate affinity are positively selected and advance to the medulla. There, they encounter dendritic cells and medullary epithelial cells presenting self-antigens. Thymocytes that bind these self-antigens too strongly are eliminated through apoptosis, a controlled cellular suicide. The surviving T cells, now mature and educated, exit the thymus and enter circulation, ready to recognize foreign antigens presented on MHC molecules without attacking normal tissues. This rigorous education is necessary because T cell receptors are generated through random genetic recombination, a process that creates enormous diversity but also produces many receptors that would be useless or actively harmful. The thymus acts as a quality control checkpoint, ensuring that only appropriate T cells are released into the body. As the thymus shrinks with age, the generation of new T cells declines, and the immune system increasingly relies on memory T cells produced earlier in life. This contributes to the age-related decline in immune function, particularly the reduced ability to respond to novel pathogens. While the primary lymphoid organs generate immune cells, the secondary lymphoid organs are where immune responses actually occur. These structures are strategically positioned throughout the body, monitoring for signs of infection and coordinating responses when threats are detected. The most numerous of these are the lymph nodes, small bean-shaped structures distributed along lymphatic vessels. An adult human has several hundred lymph nodes, clustered in particular regions: the neck, armpits, groin, chest, and abdomen. Each lymph node is encased in a fibrous capsule from which connective tissue, trabeculae, extend inward, dividing the node into compartments. Lymph enters the node through multiple afferent lymphatic vessels, percolates through the node's interior, and exits via a single efferent lymphatic vessel at the hilum. This one-way flow ensures that lymph is thoroughly filtered as it passes through. The node's interior is organized into three regions: an outer cortex rich in B cells, organized into structures called follicles, a pericortex containing T cells and dendritic cells, and an inner medulla with antibody-secreting plasma cells and lymphocytes preparing to exit. When pathogens or foreign antigens drain from tissues into lymphatic vessels, they are carried to the nearest lymph node. Here, resident dendritic cells and macrophages patrol for suspicious material, capturing and processing antigens. If a dendritic cell in the tissues encounters a pathogen, it matures, migrates to the nearest lymph node, and presents its findings to T cells in the pericortex. If a T cell bears a receptor that recognizes the presented antigen, it becomes activated, proliferates rapidly, and differentiates into effector cells that can help coordinate the immune response. B cells in the cortical follicles undergo a similar process. When a B cell's surface antibody binds to its specific antigen, and the B cell receives appropriate signals from helper T cells, it too becomes activated. The follicle transforms into a germinal center, a site of intense cellular proliferation where B cells multiply and their antibody genes undergo additional mutation and selection. This process, called affinity maturation, produces B cells with increasingly refined antibodies, better able to bind the specific antigen that triggered the response. Some of these B cells differentiate into plasma cells that secrete antibodies in large quantities. Others become memory B cells, long-lived sentinels that will respond rapidly if the same antigen is encountered again. The lymph node is designed to maximize the chance that rare antigen-specific lymphocytes will encounter their matching antigen. Lymphocytes circulate continuously, entering lymph nodes from the bloodstream via specialized blood vessels called high endothelial venules. They wander through the node's interior, surveying antigens presented by dendritic cells and macrophages. If they don't find their specific antigen, they exit via the efferent lymphatic and eventually return to the bloodstream, continuing their patrol. If they do encounter their antigen, they are retained in the node, becoming activated and proliferating before eventually leaving as effector cells ready to combat the infection. This circulation pattern means that lymphocytes are constantly surveying multiple lymph nodes, increasing the likelihood that the right cell will be in the right place at the right time. It's an elegant solution to a statistical problem. With millions of possible antigens and billions of lymphocytes, each specific for only one or a few antigens, how do you ensure that matches occur? The answer is constant movement and multiple opportunities for encounter at the lymph nodes where antigens collect. When you develop a bacterial infection, the lymph nodes near the site often become swollen and tender, sometimes palpable under the skin. This swelling reflects the intense cellular activity within lymphocytes, proliferating by the millions, plasma cells secreting antibodies, and increased blood flow, bringing more cells and resources. The swollen nodes are a sign that the immune response is working, though they can certainly be uncomfortable. Once the infection is cleared, the nodes gradually return to their normal size, though they retain memory cells that will respond faster if the same pathogen returns. The spleen, the largest lymphoid organ, sits in the upper left abdomen, tucked beneath the rib cage. Unlike lymph nodes, which filter lymph, the spleen filters blood, removing old or damaged red blood cells, recycling their components, and surveying for blood-borne pathogens. A healthy adult spleen weighs about 150 to 200 grams and processes roughly half a liter of blood per minute, an impressive throughput for an organ of its size. The spleen's interior divides into two functionally distinct regions, the red pulp and the white pulp. The red pulp, which gives the spleen its characteristic color, is involved in filtering blood. Macrophages here remove old red blood cells, capturing them in a maze of sinusoids and breaking them down. Iron from hemoglobin is salvaged and recycled. Bilirubin is sent to the liver for disposal, and the cellular components are broken down. Platelets are also stored in the red pulp, available for rapid release if bleeding occurs. The white pulp serves immune functions. It consists of lymphoid tissue surrounding arterioles, forming periarteriolar lymphoid sheaths rich in T cells with adjacent B cell follicles. When blood-borne antigens arrive, they are captured by antigen-presenting cells in the white pulp and displayed to lymphocytes. Immune responses to blood infections are often initiated here, with the spleen playing a particularly important role in responding to encapsulated bacteria like Streptococcus pneumoniae. People who have had their spleen removed, whether due to trauma or disease, face increased risk of severe bacterial infections, particularly from these encapsulated organisms. Beyond lymph nodes and spleen, the body maintains numerous smaller collections of lymphoid tissue in strategic locations. The tonsils, rings of lymphoid tissue in the throat, monitor the oral and nasal passages for pathogens. The palatine tonsils, the ones most people think of when they hear the word, sit on either side of the back of the throat. The pharyngeal tonsil, or adenoids, occupies the posterior wall of the nasopharynx. The lingual tonsils sit at the base of the tongue. Together, these form Waldeier's ring, a defensive barrier screening the entrance to the respiratory and digestive tracts. Peyer's patches, collections of lymphoid follicles in the small intestine, perform similar surveillance in the gut. The intestinal lining faces an enormous challenge. It must absorb nutrients while maintaining a barrier against the trillions of bacteria inhabiting the intestinal lumen. Peyer's patches monitor the gut contents, sampling antigens that cross the intestinal epithelium through specialized cells called M cells. This allows the immune system to survey the intestinal contents and respond to pathogenic bacteria while maintaining tolerance to food antigens and beneficial microbiota. The appendix, long dismissed as a vestigial organ, has recently been recognized as an immunological structure. It contains substantial lymphoid tissue and may serve as a reservoir for beneficial gut bacteria, repopulating the intestines after diarrheal illnesses clear out the normal microbiota. While not essential, the appendix contributes to immune surveillance in the gut, and its removal doesn't appear to cause significant immunodeficiency. Connecting all these organs and tissues are the lymphatic vessels, a parallel circulatory system that drains excess fluid from tissues and returns it to the bloodstream. Unlike blood vessels, which form closed loops, lymphatic vessels are open-ended. Tiny lymphatic capillaries in the tissues collect interstitial fluid, proteins that have leaked from blood vessels, cellular debris, and immune cells. This fluid, now called lymph, moves through progressively larger lymphatic vessels, passing through lymph nodes for filtering along the way, and eventually empties into the subclavian veins near the base of the neck, rejoining the bloodstream. Lymphatic vessels lack the high-pressure pump of the cardiovascular system. Instead, lymph movement depends on external forces, skeletal muscle contractions that squeeze the vessels, pressure changes in the chest during breathing, and the pulsation of nearby arteries. One-way valves in the lymphatic vessels prevent backflow, ensuring lymph moves consistently toward the heart. The vessels themselves have smooth muscle in their walls that can contract, but this provides only gentle assistance. This is why prolonged immobility can lead to fluid accumulation in the tissues, edema, as lymph drainage slows. The rate of lymph flow varies considerably, estimated at roughly 2 to 4 liters per day, returning to circulation. This might seem modest compared to the cardiovascular system's 7,000 or so liters per day, but lymphatic drainage serves critical functions beyond just fluid balance. It transports immune cells from tissues to lymph nodes, carries antigens from infection sites to where immune responses are coordinated, and removes proteins and large molecules that cannot re-enter blood capillaries directly. Damage to lymphatic vessels or blockage by parasites, tumors, or surgical removal of lymph nodes can impair drainage, causing lymphedema, persistent swelling due to accumulated lymph. This can occur after cancer surgery when lymph nodes are removed, or from parasitic infections like lymphatic filariasis, where filarial worms block lymphatic vessels. The resulting swelling can be disfiguring and predisposes to infections because immune surveillance is impaired when lymph cannot drain properly. This network of organs and vessels forms the infrastructure of immunity. The primary lymphoid organs generate diverse populations of immune cells. The secondary lymphoid organs bring these cells together with antigens, facilitating the encounters that trigger immune responses. The lymphatic vessels connect these sites, allowing cells and information to move where needed. It is an elegant distributed system, resilient because it lacks a single point of failure, effective because it maximizes the chance that the right cells will find the right threats. But organs and vessels are just the framework. To understand immunity, We must now meet the cells that actually perform immune functions, the diverse population of white blood cells that patrol, recognize, and eliminate threats to the body. The immune system's work is performed by cells, billions upon billions of them, circulating through blood and lymph, residing in tissues, and standing watch at the body's borders. These cells are collectively known as leukocytes, from the Greek words for white and cell, though in reality they are mostly colorless. The designation white distinguishes them from the far more numerous red blood cells, whose hemoglobin gives blood its characteristic color. Where red blood cells are dedicated specialists, single-minded in their transport of oxygen, white blood cells are a diverse collective, each type specialized for particular defensive functions. All blood cells, both red and white, originate from hematopoietic stem cells in the bone marrow. These remarkable cells possess two crucial properties. They can differentiate into multiple cell types, and they can self-renew, maintaining their own population while also generating progeny that become more specialized. A single hematopoietic stem cell, if transplanted into an irradiated host whose native blood cells have been destroyed, can reconstitute the entire blood and immune system. This capacity makes bone marrow transplantation possible, a treatment that can cure certain cancers and immunodeficiency disorders. The process of blood cell formation, hematopoiesis, proceeds through a series of increasingly restricted developmental stages. The hematopoietic stem cell first divides to produce either a common myeloid progenitor or a common lymphoid progenitor. This initial branching point separates two major lineages. The myeloid lineage will produce red blood cells, platelets, and several types of white blood cells, neutrophils, eosinophils, basophils, monocytes, and their tissue-resident relatives, the macrophages and dendritic cells. The lymphoid lineage produces the lymphocytes, T cells, B cells, and natural killer cells. Each subsequent division further restricts developmental potential. The common myeloid progenitor splits into a megakaryocyte erythroid progenitor, which produces platelets and red blood cells, and a granulocyte monocyte progenitor. This latter cell gives rise to the granulocytes, white blood cells characterized by prominent granules in their cytoplasm visible under the microscope. The granulocytes include neutrophils, eosinophils, and basophils, named for the staining properties of their granules, neutral, eosine-loving, and base-loving respectively. The granulocyte monocyte progenitor also produces monocytes, which leave the bone marrow and circulate briefly before entering tissues where they differentiate into macrophages or dendritic cells. The common lymphoid progenitor, by contrast, generates the lymphocytes. Some of these cells complete their development in the bone marrow, becoming B lymphocytes. Others leave the marrow as immature cells and migrate to the thymus, where they become T lymphocytes. A third population develops into natural killer cells, part of the innate immune system despite their lymphoid origin. These developmental pathways are not rigidly fixed. Recent research has revealed considerable plasticity, with some cells capable of switching lineages under certain conditions, but the general scheme holds. Myeloid and lymphoid progenitors giving rise to the diverse cast of immune cells. This developmental process is tightly regulated by growth factors and cytokines, signaling molecules that promote proliferation and differentiation of specific cell types. During infection, the demand for neutrophils increases dramatically, and the bone marrow responds by ramping up production. Colony-stimulating factors like GCSF, granulocyte colony-stimulating factor, drive this increased output. Physicians sometimes administer GCSF to patients undergoing chemotherapy, which destroys rapidly dividing cells, including bone marrow stem cells, to help reconstitute their white blood cell populations more quickly. The use of such factors illustrates how deeply we've come to understand and manipulate the processes that generate immune cells. Let us meet the cells themselves, starting with the most abundant, the neutrophils. These cells account for 50 to 70% of circulating white blood cells in healthy adults, a proportion that increases dramatically during bacterial infections. A cubic millimeter of blood contains roughly 4,000 to 11,000 white blood cells total, meaning neutrophil counts typically range from 2,000 to 7,700 per cubic millimeter. Neutrophils are phagocytes, professional eaters of microbes, and they are the first responders to sites of infection or tissue damage. Structurally, a neutrophil is 12 to 15 micrometers in diameter with a distinctive multi-lobed nucleus, typically displaying 3 to 5 lobes connected by thin strands of nuclear material. This segmented nucleus gives neutrophils an alternative name, polymorphonuclear leukocytes, often abbreviated PMNs. The cytoplasm contains numerous granules packed with antimicrobial substances, enzymes like myeloperoxidase that generate reactive oxygen species, proteases that degrade bacterial proteins, and antimicrobial peptides called defensins that punch holes in microbial membranes. Neutrophils live fast and die young. Newly produced in the bone marrow, they circulate for only 6 to 8 hours before migrating into tissues, where they survive another day or two at most. This short lifespan means the bone marrow must produce about 100 billion neutrophils daily just to maintain steady-state numbers. During infection, production increases many-fold, and immature neutrophils called band cells may be released into circulation prematurely, a sign that the body is under stress. The presence of band cells, identified by their less segmented nuclei, is sometimes called a left shift in reference to older schemes for reporting complete blood counts and indicates active infection or inflammation. When neutrophils detect signs of infection, chemical signals called chemokines, they exit the bloodstream in a carefully choreographed process. First, they roll along the endothelial cells lining blood vessels, weakly adhering through selectin molecules. If the chemokine gradient is strong enough, the neutrophil activates integrins, stronger adhesion molecules that firmly attach it to the endothelium. The cell then squeezes between endothelial cells in a process called diapedesis, migrating into the tissue toward the infection site. Once there, neutrophils phagocytose a bacteria, engulfing them in internal compartments called phagosomes and destroying them with the contents of their granules. Neutrophils can also release their DNA and granular contents extracellularly, forming neutrophil extracellular traps, or dot nets. These sticky webs of chromatin, decorated with antimicrobial proteins, trap bacteria, preventing their spread and killing them. This process, enetosis, is essentially a form of cellular suicide, trading the neutrophil's life to contain infection. The formation of nets can be beneficial in limiting pathogen spread, but excessive net formation contributes to thrombosis and tissue damage, reminding us that immune mechanisms, while protective, can also cause harm. Eosinophils, the eosine-loving granulocytes, are far less abundant than neutrophils, accounting for only 1 to 4% of circulating white blood cells. They are 12 to 17 micrometers in diameter, with a characteristic bilobed nucleus and large cytoplasmic granules that stain bright red-orange with eosin dye. These granules contain toxic proteins, including major basic protein, eosinophil cationic protein, and eosinophil peroxidase, which are particularly effective against parasites too large to phagocytose. Eosinophils play a crucial role in defense against helminthic worms, multicellular parasites that cannot be simply engulfed and digested. When antibodies coat a worm, eosinophils bind to these antibodies via Fc receptors and discharge their granular contents directly onto the parasite's surface, damaging its outer tegument. This antibody-dependent cell-mediated cytotoxicity provides an effective, if somewhat indiscriminate, means of attacking large parasites. Unfortunately, eosinophils also contribute to allergic inflammation, particularly in asthma, where their products damage airway epithelium. Elevated eosinophil counts in blood or tissues often indicate either parasitic infection or allergic disease. Basophils, the least common granulocytes, represent less than 1% of circulating white blood cells. At 10 to 14 micrometers in diameter, they are similar in size to neutrophils, but distinguished by their large basophilic granules that stain dark blue or purple. These granules contain histamine, heparin, and other vasoactive substances. When activated, basophils degranulate, releasing these contents and triggering local inflammation, vasodilation, increased vascular permeability, and smooth muscle contraction. Basophils function similarly to mast cells, tissue-resident cells that are actually distinct despite their functional overlap. Both cell types bear high-affinity receptors for IgE antibodies, and when antigens cross-link these bound antibodies, the cells degranulate. This mechanism underlies immediate hypersensitivity reactions, the allergic responses that occur within minutes of allergen exposure. Basophil activation causes the itching, swelling, and respiratory distress characteristic of allergies, from hay fever to life-threatening anaphylaxis. While this seems maladaptive in modern environments, the Ig-E basophil mast cell axis likely evolved to combat parasites and remains useful in that context, even if it misfires against pollen and peanuts. Monocytes represent 2 to 8% of circulating white blood cells. These are large cells, 15 to 30 micrometers in diameter, with kidney-shaped or horseshoe-shaped nuclei and abundant cytoplasm. Unlike granulocytes with their short circulatory lifespans, monocytes circulate for several days, but the blood is merely a transport phase for these cells. Their true purpose is revealed when they migrate into tissues and differentiate into macrophages or dendritic cells, two of the most important cell types in immunity. Macrophages, whose name literally means big eaters, are long-lived tissue-resident phagocytes. Depending on the tissue they inhabit, they acquire specific names and specialized functions. Kupffer cells in the liver filter blood from the digestive tract, removing bacteria that cross the intestinal barrier. Alveolar macrophages in the lungs clear inhaled particles and pathogens. Microglia in the brain maintain neural tissue and respond to central nervous system infections or injury. Osteoclasts in bone, while primarily involved in bone remodeling, are also macrophage relatives. Despite these diverse locations and functions, all share the basic macrophage program: phagocytosis, antigen presentation, and cytokine secretion. Macrophages are professional phagocytes capable of engulfing and destroying a remarkable variety of materials: bacteria, fungi, parasites, dead cells, cellular debris, even inorganic particles like asbestos fibers or tattoo ink. They recognize targets through various receptors that bind to patterns common on pathogens but absent from host cells, called pathogen associated molecular patterns, or PAMPs. These include bacterial cell wall components like lipopolysaccharide, viral nucleic acids, and fungal cell wall components like beta glucans. Upon binding these patterns, macrophages extend pseudopodia around the target, engulfing it in a membrane-bound phagosome. The phagosome then fuses with lysosomes, organelles packed with hydrolytic enzymes and toxic chemicals. The resulting phagolysosome is an intensely hostile environment. Acidic pH, reactive oxygen species, reactive nitrogen species, antimicrobial peptides, and enzymes that tear apart proteins, nucleic acids, and carbohydrates. Most bacteria cannot survive this onslaught and are broken down into their component parts. Some pathogens, however, have evolved mechanisms to resist or escape. Mycobacterium tuberculosis prevents phagosome-lysosome fusion. Listeria monocytogenes escapes from the phagosome into the cytoplasm, and Leishmania parasites are actually adapted to survive in the harsh phagolysosomal environment. Beyond phagocytosis, macrophages serve as antigen-presenting cells, displaying fragments of ingested material on their surface for recognition by T cells. This bridges innate and adaptive immunity, with macrophages providing information about what they've encountered to the cells of the adaptive system. Macrophages also secrete numerous cytokines, chemical messengers that recruit other immune cells, activate inflammation, and influence the differentiation of other cell types. A macrophage, encountering bacteria, secretes tumor necrosis factor alpha and interleukin-1, pro-inflammatory cytokines that activate endothelial cells, recruit neutrophils, and trigger fever. Different stimuli cause macrophages to secrete different cytokine profiles, tailoring the immune response to the specific threat. Dendritic cells, the other major differentiation product of monocytes, are specialized for antigen presentation. They are named for their dendrite-like protrusions, cytoplasmic extensions that give them a branched, almost neural appearance under the microscope. Immature dendritic cells reside in tissues, particularly at barrier sites like the skin, gut, and respiratory tract, where they constantly sample their surroundings through phagocytosis and macropinocytosis, drinking in extracellular fluid and any dissolved antigens. When a dendritic cell encounters signs of infection, PAMPs, damage-associated molecular patterns from injured cells, or inflammatory cytokines, it matures and migrates to the nearest lymph node. During maturation, the cell reduces its phagocytic activity and upregulates surface molecules needed for T cell activation. It processes its captured antigens, loading fragments onto MHC molecules and displaying them on its surface. In the lymph node, the mature dendritic cell presents these antigens to T cells. If a T cell bears a receptor that recognizes the displayed antigen, and if the dendritic cell provides appropriate co-stimulatory signals, the T cell becomes activated. This makes dendritic cells, arguably, the most important antigen-presenting cells in the immune system. While macrophages and B cells can also present antigens, dendritic cells are uniquely efficient at activating naive T cells, those that have never encountered their specific antigen. The term for this function is professional antigen presentation, and it is the crucial link between innate detection of threats and adaptive immune responses. A dendritic cell presenting viral antigens can activate antiviral T cells. One presenting tumor antigens might activate anti-tumor immunity. The dendritic cell is the messenger that tells adaptive immunity what to target. Now we turn to the lymphocytes, cells of the adaptive immune system characterized by their specificity and memory. Lymphocytes are smaller than most other leukocytes, typically 7 to 12 micrometers in diameter, with large nuclei occupying most of the cell volume and a thin rim of cytoplasm. Despite their morphological similarity under the microscope, lymphocytes are functionally diverse, divided into several major populations. B lymphocytes, as mentioned earlier, mature in the bone marrow and are responsible for antibody production. Each B cell bears surface immunoglobulins, antibody molecules that serve as receptors. A given B cell expresses roughly 100,000 copies of the same antibody on its surface, all with identical antigen binding sites. When the B cell encounters an antigen that binds to this receptor and receives appropriate signals from helper T cells, it becomes activated. The activated B cell proliferates, generating a large clone of identical cells, a process called clonal expansion. Most of these proliferating B cells differentiate into plasma cells, antibody factories that secrete thousands of antibody molecules per second. Plasma cells are morphologically distinct, larger than naive B cells, with abundant rough endoplasmic reticulum, the cellular machinery for protein synthesis, giving their cytoplasm a characteristic appearance. They live only a few days, but produce enormous quantities of antibodies during their brief existence. These secreted antibodies circulate in blood and lymph and permeate into tissues, binding to their specific antigen wherever they encounter it. Some activated B cells, instead of becoming plasma cells, differentiate into memory B cells. Some activated B cells, instead of becoming plasma cells, differentiate into memory B cells. These long-lived cells persist for years, sometimes decades, maintaining the capacity to rapidly respond if the same antigen is encountered again. Memory B cells are the basis of long-lasting immunity, the reason a single infection or vaccination can provide protection for a lifetime. They circulate through blood and lymphoid tissues, surveying for their specific antigen. Upon re-encounter, they activate much more rapidly than naive B cells, differentiating into plasma cells within days and producing high levels of high-affinity antibodies, often preventing illness entirely, or at least reducing its severity. T lymphocytes, educated in the thymus, are divided into several functionally distinct subsets based on their surface markers and roles. The two major classes are CD4+ and CD8+ T cells, named for the co-receptor molecules they express. These co-receptors determine which type of MHC molecule the T cell can recognize, and thus what kind of antigens they respond to. CD8+ T cells, also called cytotoxic T lymphocytes or killer T cells, recognize antigens presented on MHC class I molecules. Since all nucleated cells express MHC class I and use it to display samples of their internal proteins, CD8+ T cells can survey any cell in the body for signs of infection or abnormality. A virus-infected cell displays viral peptides on its MHC class I molecules. A cancer cell might display mutated peptides. When a CD8+ T cell recognizes such an abnormal peptide, it becomes activated and kills the presenting cell. Cytotoxic T cells kill through several mechanisms. They can secrete perforin, a protein that forms pores in the target cell membrane, and granzymes, proteases that enter through these pores and trigger apoptosis. They can also use Fas ligand, binding to Fas receptors on the target cell and activating the cell's suicide machinery directly. These mechanisms allow CD8 plus T cells to eliminate infected or cancerous cells with precision, though some collateral damage to nearby cells is inevitable. The death of infected cells prevents viruses from completing their replication cycle, and the clearance of cancer cells prevents tumor formation. CD4 plus T cells, often called helper T cells, recognize antigens presented on MHC class II molecules. These molecules are expressed only on professional antigen-presenting cells, dendritic cells, macrophages, and B cells. This restriction means CD4 plus T cells primarily encounter antigens that have been phagocytosed from the extracellular environment. When a CD4 plus T cell recognizes its specific antigen presented on MHC class II, it becomes activated and differentiates into one of several helper T cell subtypes, each specialized for different types of immune responses. Th1 cells secrete interferon gamma and tumor necrosis factor, cytokines that activate macrophages and promote cell-mediated immunity against intracellular pathogens. Two cells produce interleukin-4, interleukin-5, and interleukin-1-3, supporting antibody production, especially IgE, and activating eosinophils, useful against parasites, but also responsible for allergic inflammation. Th17 cells secrete interleukin-1-7 and promote neutrophil recruitment, important for defense against extracellular bacteria and fungi but implicated in autoimmune diseases. Regulatory T cells, or TREGs, produce interleukin-10 and TGF-beta, suppressing immune responses and maintaining tolerance to self-antigens. This functional diversity among CD4 plus T cells allows the immune system to tailor its response to different types of threats. The differentiation pathway is influenced by the cytokines present during T cell activation, creating a feedback system where the innate immune response shapes the subsequent adaptive response. A bacterial infection triggering interleukin 1-2 secretion from dendritic cells will push CD4 plus T cells toward the Th1 phenotype. A parasitic infection with IL-4 inches, the environment favors two differentiation. Natural killer cells, or NK cells, represent a third lymphocyte population. Despite their lymphoid origin, they function as part of innate immunity, responding within hours rather than days, and lacking the exquisite specificity of T and B cells. NK cells are 10 to 15 micrometers in diameter, with prominent cytoplasmic granules containing perforin and granzymes. They circulate through blood and lymphoid tissues, surveying other cells for signs of infection or malignancy. NK cells recognize targets through a balance of activating and inhibitory receptors. Inhibitory receptors recognize MHC class I molecules, the same molecules that present antigens to CD8 plus T cells. When an NK cell encounters a healthy cell expressing normal levels of MHC class I, the inhibitory signals predominate and the target is spared. Many viruses and some tumors downregulate MHC class I expression to avoid CD8 plus T cell recognition. Activating receptors on NK cells recognize stress-induced molecules that appear on infected or transformed cells. When activating signals outweigh inhibitory signals, the NK cell releases its granular contents, killing the target through the same perforin and granzyme mechanism used by CD8 plus T cells. NK cells also express Fc receptors that bind to antibodies, allowing them to perform antibody dependent cell mediated cytotoxicity, killing antibody coated targets. This provides a bridge between adaptive immunity, which produces specific antibodies, and innate immunity, which provides the killer cells. This cellular caste, from neutrophils to NK cells, represents the immune system's workforce. Each cell type has specialized functions, yet they coordinate through an elaborate communication network. Neutrophils respond first, but recruit monocytes that become macrophages. Dendritic cells activate T cells, which help B cells produce antibodies, which in turn enhance phagocytosis by neutrophils and macrophages. The system is deeply interconnected, with redundancy built in at every level. No single cell type is essential, as demonstrated by individuals with deficiencies in specific populations who, while more susceptible to certain infections, can often survive with remaining immune functions, compensating partially. Understanding these cells and their origins is the foundation for understanding immunity, but cells do not function in isolation. They communicate through a complex chemical language of cytokines and other signaling molecules, coordinating their activities to mount effective immune responses while limiting damage to healthy tissue. It is to this chemical communication system that we now turn our attention. The immune system's cellular components would be ineffective without mechanisms for communication. Individual cells must coordinate their activities, recruit reinforcements, activate or suppress responses, and share information about threats. This coordination occurs through chemical messengers, small proteins secreted by cells that bind to receptors on other cells, altering their behavior. These messengers, collectively called cytokines, form an elaborate communication network that is essential for effective immunity. The term cytokine, from Greek roots meaning cell and movement, encompasses a diverse array of signaling proteins. Chemokines attract cells to specific locations, creating gradients that direct cellular migration. Interleukins, originally thought to mediate communication between leukocytes, are numbered sequentially as they are discovered, currently extending past IL-37, though the numbering is incomplete, with some numbers unassigned. Interferons got their name from their ability to interfere with viral replication. Colony-stimulating factors promote the proliferation and differentiation of blood cell precursors. Tumor necrosis factors were named for their ability to cause tumor regression in early experiments, though their functions extend far beyond this. These categorizations are historical and somewhat arbitrary. Many cytokines have pleiotropic effects, influencing multiple cell types in different ways. Many are also redundant, with several cytokines capable of inducing similar responses, a built-in insurance policy ensuring that no single signaling pathway is absolutely essential. This redundancy complicates research, making it difficult to assign definitive functions to individual cytokines, but it provides robustness to the immune system, allowing it to function even when specific components are defective. Cytokines typically act at very low concentrations, often in the picomolar to nanomolar range, yet produce profound biological effects. They bind to specific receptors on target cells, triggering intracellular signaling cascades that alter gene expression, cellular metabolism, proliferation, differentiation, or survival. A single cytokine binding to its receptor can initiate a cascade that activates thousands of molecules inside the cell, amplifying the original signal. This amplification allows rare events, like a single dendritic cell encountering a pathogen to have systemic consequences. The actions of cytokines can be autocrine, affecting the cell that secreted them, paracrine, affecting nearby cells, or endocrine, affecting distant cells after traveling through the bloodstream. Most cytokine actions are autocrine or paracrine, working locally to coordinate responses in the immediate vicinity of the threat. However, certain cytokines, particularly those involved in systemic responses like fever, do circulate and act at a distance. The distinction is not absolute, as local cytokine secretion during severe infections can spill over into systemic circulation, producing body-wide effects. Interleukins, the most numerous category, mediate diverse functions. Interleukin 1, one of the earliest discovered, is produced by macrophages and epithelial cells in response to infection or tissue damage. It has numerous effects, promoting fever by acting on the hypothalamus, activating endothelial cells to support immune cell recruitment, stimulating acute phase protein synthesis in the liver, and promoting T cell activation. IL-1 exists in two main forms, IL-1 alpha and IL-1 beta, both binding the same receptor but differing in regulation and processing. IL-1 beta is the more studied form, synthesized as an inactive precursor that requires cleavage by the enzyme caspase-1 to become active, a process that occurs within multi-protein complexes called inflammasomes. Interleukin 2, secreted by activated T cells, is crucial for T cell proliferation. It acts primarily in an autocrine manner, with T cells secreting IL-2 and responding to it themselves, driving their own expansion. IL-2 also promotes the survival and function of regulatory T cells, contributing to immune regulation. The importance of IL-2 is evident in its therapeutic applications. Recombinant IL-2 has been used to treat certain cancers by stimulating T cell responses against tumors, though the treatment can cause significant side effects due to widespread immune activation. Interleukin 4, produced mainly by two cells, mast cells, and basophils, is a central regulator of the Th2 type response. It promotes B cell activation, antibody class switching to IgE, and the differentiation of naive CD 4 plus T cells into Th2 cells, creating a positive feedback loop. IL-4 also activates eosinophils and promotes alternative activation of macrophages, a state associated with tissue repair but also with allergic inflammation. The IL-4 signaling pathway is a target for therapeutic intervention in allergic diseases, with antibodies that block IL-4 or its receptor showing efficacy in treating conditions like atopic dermatitis. Interleukin 6, produced by many cell types, including macrophages, T cells, and fibroblasts, is a pleiotropic cytokine with both pro- and anti-inflammatory properties. It stimulates acute phase protein synthesis, induces fever, promotes B cell maturation into antibody-secreting plasma cells, and supports the differentiation of Th17 cells. IL-6 levels are elevated in many inflammatory and autoimmune diseases, and blocking IL-6 signaling has proven effective in treating rheumatoid arthritis and other conditions. The dual nature of IL-6, beneficial in acute infections but harmful when chronically elevated, illustrates the complexity of cytokine biology. Interleukin 10 is predominantly anti-inflammatory, produced by regulatory T cells, macrophages, and other cell types to limit immune responses and prevent excessive tissue damage. It inhibits the production of pro-inflammatory cytokines like IL-1, TNF alpha, and IL-12, and suppresses antigen presentation and T cell activation. IL-10 is crucial for maintaining intestinal homeostasis, preventing inappropriate inflammation in response to commensal gut bacteria. Some pathogens exploit this, inducing IL-10 production to dampen immune responses and facilitate their own survival. Interleukin 1-2, produced by dendritic cells and macrophages, is a key driver of cell-mediated immunity. It promotes the differentiation of naive CD4 plus T cells into Th1 cells and enhances NK cell and CD8 plus T cell cytotoxicity. IL-12 is particularly important in responses to intracellular pathogens like mycobacteria and certain viruses. Genetic defects in IL-12 production or signaling cause severe susceptibility to mycobacterial infections, demonstrating its critical role. The cytokine's potent immune-stimulating properties have led to its investigation as a cancer immunotherapy, though systemic administration causes significant toxicity. Interleukin 1-7, the signature cytokine of Th17 cells, promotes inflammation, particularly neutrophil recruitment. It stimulates the production of chemokines that attract neutrophils and induces the expression of antimicrobial peptides. IL-17 is important for defense against extracellular bacteria and fungi, especially at mucosal surfaces. However, excessive IL-17 signaling contributes to autoimmune diseases, including psoriasis, rheumatoid arthritis, and inflammatory bowel disease. Therapies targeting IL-17 or its receptor have proven effective in treating these conditions, though they can increase susceptibility to certain infections. The interferons constitute another major cytokine family. Type 1 interferons, primarily IFN alpha and IFN beta, are produced by most cell types in response to viral infection. When a cell detects viral nucleic acids through pattern recognition receptors, it secretes type 1 interferons, which bind to receptors on neighboring cells, inducing an antiviral state. Interferon signaling activates hundreds of genes, interferon-stimulated genes, many of which inhibit various stages of viral replication. Some interfere with viral protein synthesis, others degrade viral RNA, still others inhibit viral assembly or release. Different chemokines attract different cell types. CXCL8, also called IL-8, primarily attracts neutrophils and is crucial for their recruitment to infection sites. CCL2 attracts monocytes, CCL5 attracts T cells, and CCL11 attracts eosinophils. This specificity allows the immune system to recruit appropriate cells to different types of infections. A bacterial infection triggering CXCL8 production will recruit neutrophils, effective against bacteria. A parasitic infection inducing CCL11 brings in eosinophils specialized for anti-parasite responses. Chemokines also direct lymphocyte trafficking through lymphoid organs. CCL19 and CCL21 attract naive T cells into lymph nodes, where they can encounter antigen-presenting dendritic cells. CXCL13 attracts B cells into B cell follicles. This choreographed movement ensures that immune cells are in the right place at the right time, maximizing the efficiency of immune responses. Defects in chemokine receptors can cause immunodeficiency, as immune cells cannot migrate properly to sites of infection or to lymphoid organs. The complexity of cytokine networks creates challenges for understanding and manipulating immunity. Individual cytokines rarely act alone. They function in combinations, often with synergistic or antagonistic interactions. IL-1 and TNF alpha synergize to activate endothelial cells more strongly than either alone. IL-10 antagonizes the effects of IL-12, balancing Th1 and regulatory responses. The timing and sequence of cytokine exposure matter. Dendritic cells exposed to different cytokines at different times during maturation develop distinct properties that influence subsequent T cell activation. This complexity gives rise to the concept of cytokine storms, pathological states where positive feedback loops drive excessive cytokine production. This can occur in severe infections, particularly influenza and emerging viral diseases like COVID-19, and in some cancer immunotherapies. The massive release of pro-inflammatory cytokines causes systemic inflammation, vascular leak, tissue damage, and multi-organ failure. The very immune responses meant to protect become the primary threat. Understanding and preventing cytokine storms is an active area of research, with therapeutic strategies focused on blocking specific cytokines or breaking the positive feedback loops. Cytokine biology also explains many drug side effects and disease symptoms. The fatigue, muscle aches, and fever characteristic of influenza are not caused directly by the virus, but by the cytokines produced in response to infection. IL-1, IL-6, and TNF alpha all contribute to these systemic symptoms. The side effects of immunotherapy, whether interferon treatment or checkpoint inhibitor antibodies for cancer, largely reflect cytokine-mediated immune activation. Even the placebo effect may involve cytokines, as psychological factors can influence cytokine production, demonstrating the intimate connection between nervous and immune systems. Therapeutic manipulation of cytokine networks has become a cornerstone of modern medicine. Beyond the TNF inhibitors and IL-6 blockers already mentioned, numerous other cytokine targeting therapies have been developed. IL-1 inhibitors treat autoinflammatory syndromes and gouty arthritis. IL-4 and IL-5 inhibitors treat severe asthma and eosinophilic disorders. IL-17 inhibitors treat psoriasis. IL-2 is used to expand T cell populations in cancer therapy. Interferons treat viral infections and certain cancers. GM-CSF is used to accelerate neutrophil recovery after chemotherapy. The list continues to grow as our understanding deepens. Yet for all our sophistication in measuring and manipulating cytokines, much remains mysterious. The same cytokine can have different, even opposite effects depending on concentration, cellular context, and what other signals are present. The kinetics matter. A brief pulse of cytokine exposure produces different outcomes than sustained exposure. Local versus systemic effects differ dramatically, and individual genetic variation in cytokine genes, receptors, and signaling pathways means that people respond differently to the same infections and treatments. The chemical language of immunity is both specific and flexible, allowing precise coordination while maintaining robustness through redundancy. Cytokines link innate and adaptive immunity, translate microbial detection into appropriate responses, recruit the right cells to the right locations, and scale responses to match threat severity. They are the invisible threads connecting the cellular cast we met in the previous chapter, transforming individual actors into a coordinated defense system. With this understanding of cellular components and chemical communication, we can now examine the immune system's first line of defense, the physical and chemical barriers that prevent most pathogens from ever entering the body's internal environment. Before any immune cell is called to action, before any cytokine is secreted, before adaptive immunity even becomes aware of a threat, the body deploys a formidable array of passive defenses. These are the barriers, mechanical obstacles that physically prevent pathogen entry, chemical agents that neutralize or destroy microbes, and ecological communities of beneficial organisms that outcompete potential pathogens. These defenses are often categorized as innate immunity, but they are more accurately understood as pre-immunity, the fortifications that must be breached before the immune system proper becomes engaged. The skin, our most obvious barrier, is a remarkable organ, covering approximately 1.5 to 2 square meters in adults and weighing roughly 15% of body weight. It is both extensive and substantial. The skin is a multi-layered structure with the outer epidermis composed of stratified squamous epithelium, cells arranged in overlapping sheets like shingles on a roof. The outermost layer, the stratum corneum, consists of dead, flattened keratinocytes that have undergone keratinization, filling with tough keratin proteins and losing their nuclei and organelles. These dead cells, though lifeless, provide formidable protection. Keratin is highly resistant to abrasion, water, and most chemicals. The cells are cemented together with lipids that make the skin water repellent, preventing both moisture loss and pathogen entry. The stratum corneum is typically 10 to 20 cell layers thick on most body surfaces, but can exceed 100 layers on the palms and soles, reflecting the mechanical stresses these areas endure. This dead layer is constantly shed, with the entire epidermis replaced roughly every 28 days. Pathogens adhering to the surface are simply sloughed off before they can establish infection. Beneath the epidermis lies the dermis, a thicker layer of connective tissue containing blood vessels, nerve endings, hair follicles, and glands. The dermis provides structural support and nutrients to the epidermis above, and houses immune cells, particularly macrophages and dendritic cells, ready to respond if the epithelial barrier is breached. The skin's immune components, sometimes called skin-associated lymphoid tissue or SALT, include Langerhans cells, specialized dendritic cells in the epidermis that capture antigens and migrate to lymph nodes if they detect signs of infection. The skin's protective functions extend beyond its physical structure. Sebaceous glands secrete sebum, an oily mixture of lipids and cellular debris that moisturizes skin and hair while also creating a slightly acidic environment, typically pH 4 to 6. Inhospitable to many bacteria, sebum contains fatty acids with antimicrobial properties. Sweat glands contribute additional protection. Eccrine sweat contains lysozyme, an enzyme that cleaves bacterial cell walls, and antimicrobial peptides called defensins and catholycidins that directly kill bacteria and fungi. The skin also hosts a vast microbiome, estimated at 1 million bacteria per square centimeter on average, with higher densities in moist areas like the armpits and lower densities in dry areas. These resident microbes are predominantly beneficial, occupying niches that might otherwise be colonized by pathogens. They compete for nutrients, produce metabolites that inhibit pathogenic species, and stimulate the skin's immune defenses, keeping them primed without causing inflammation. The relationship is mutualistic. The skin provides habitat and nutrients. The microbes provide protection. Disrupting this ecosystem with excessive antibiotic use or aggressive cleaning can paradoxically increase susceptibility to skin infections. Where the skin ends, mucous membranes begin. These moist epithelial surfaces line body cavities that open to the exterior, the respiratory, gastrointestinal, and urogenital tracts. Unlike skin, mucous membranes cannot rely on desiccation and a thick keratinized barrier. Instead, they deploy mucus, a viscous gel composed of water, glycoproteins called mucins, salts, and various antimicrobial substances. Mucus serves multiple functions. It traps particles and microbes, preventing their adherence to epithelial cells. It lubricates surfaces, facilitating the passage of materials, and it contains antimicrobial agents that neutralize or kill trapped pathogens. The respiratory tract exemplifies these barrier mechanisms. Air entering the nose encounters a series of defenses. Coarse hairs in the nostrils trap large particles. The nasal cavity's convoluted passages, the turbinates, create turbulent airflow that causes particles to impact the mucus-coated epithelium. This mucus is continuously produced by goblet cells scattered throughout the epithelium and by submucosal glands. Trapped particles and microbes are moved by cilia, microscopic hair-like projections on epithelial cells that beat in coordinated waves, creating a current that propels mucus toward the pharynx, where it is swallowed or expectorated. This mucociliary escalator operates continuously, sweeping 10 to 100 milliliters of mucus per day out of the respiratory tract. Each ciliated cell bears approximately 200 cilia that beat 10 to 20 times per second in a coordinated pattern, moving mucus at speeds of 4 to 20 millimeters per minute. The cilia beat more rapidly in the power stroke, pushing mucus forward, and bend during the recovery stroke, slipping back underneath the mucus layer without pulling it backward. This asymmetric beating pattern requires precise coordination among adjacent cells, achieved through hydrodynamic coupling and signaling between cells. Respiratory mucus is not merely sticky, it is actively antimicrobial. It contains lysozyme at concentrations of approximately 20 micrograms per milliliter, sufficient to damage many bacterial cell walls. Lactoferrin, an iron-binding protein, is present at 5 to 50 micrograms per milliliter and sequesters iron, depriving bacteria of this essential nutrient. Defensins and other antimicrobial peptides directly kill bacteria by disrupting their membranes. Secretory IgA, the predominant antibody in mucosal secretions, binds to pathogens and prevents their adherence to epithelial cells, facilitating their removal in the mucus flow. The respiratory tract also employs mechanical clearance mechanisms beyond mucociliary action. Coughing can expel air at speeds exceeding 100 miles per hour, forcibly ejecting mucus and any trapped material. Sneezing serves a similar function for the nasal passages. These reflexes are triggered by irritant receptors that detect mechanical stimulation, chemical irritants, or inflammatory mediators released during infection. While socially awkward, they are highly effective at pathogen removal, which explains why suppressing them with medications during illness may prolong recovery. The gastrointestinal tract faces perhaps the greatest pathogen burden of any body system, with trillions of bacteria residing in the intestinal lumen and constant exposure to ingested microbes. The barriers here are multi-layered and formidable. It begins in the mouth with saliva, produced at approximately 0.5 to 1.5 liters per day by salivary glands. Saliva contains lysozyme, lactoferrin, secretory IgA, and various peroxidases with antimicrobial activity. It also dilutes and washes away potential pathogens, and its flow increases during eating, providing mechanical clearance. The stomach provides one of the body's most hostile environments for pathogens, gastric acid. Parietal cells in the gastric mucosa secrete hydrochloric acid, maintaining the stomach's pH between 1.5 and 3.5, levels that denature proteins, disrupt membranes, and kill most bacteria. The stomach also secretes pepsinogen, activated to the protease pepsin by acid, which degrades bacterial proteins. Few bacteria can survive this acidic assault. Helicobacter pylori is a notable exception, having evolved mechanisms to neutralize acid in its immediate vicinity and burrow into the protective mucus layer. The intestines present a different challenge. They must absorb nutrients while maintaining a barrier against the vast microbial population in the lumen. The intestinal epithelium is a single cell layer thick, yet remarkably effective at selective permeability. Tight junctions between epithelial cells prevent most bacteria from passing between cells. The epithelium is covered by a mucus layer, particularly thick in the colon, that physically separates bacteria from epithelial cells. This mucus is stratified, an inner layer firmly attached to the epithelium and normally sterile, and an outer layer where commensal bacteria reside. Intestinal epithelial cells secrete antimicrobial peptides, including defensins and cathelicidins. Paneth cells, specialized epithelial cells at the base of intestinal crypts, produce particularly high levels of antimicrobial peptides that help sterilize the crypt environment, protecting the intestinal stem cells located there. The epithelium is rapidly renewed, with cells shed every 3 to 5 days, preventing pathogens from establishing permanent adhesion. This rapid turnover requires a remarkable rate of cell division, with intestinal crypts serving as the proliferative zones. The intestinal microbiome represents the most sophisticated barrier mechanism. The colon alone contains approximately 14 bacteria, outnumbering human cells in the body and encoding 100-fold more genes than the human genome. These bacteria occupy virtually all available ecological niches, consuming available nutrients and producing metabolites that inhibit pathogenic species. They also stimulate the intestinal immune system, promoting the production of secretory IgA and maintaining the epithelial barrier function. Certain commensal bacteria produce bacteriocins, antimicrobial peptides that kill competing bacterial species. Others produce short-chain fatty acids through fermentation of dietary fiber. These fatty acids nourish intestinal epithelial cells and have anti-inflammatory properties. The microbiome trains the developing immune system, helping establish tolerance to harmless antigens while maintaining responsiveness to pathogens. Mice raised in sterile environments without any microbiome develop severely impaired immunity, demonstrating the microbiome's crucial role. Disruption of the intestinal microbiome, dysbiosis, can have serious consequences. Antibiotic treatment kills not only pathogenic bacteria, but also beneficial commensals, allowing opportunistic pathogens like Clostridioides difficile to proliferate unchecked. C. Difficile produces toxins that damage the intestinal epithelium, causing potentially fatal colitis. Fecal microbiota transplantation, transferring intestinal bacteria from healthy donors to patients with recurrent C. Difficile infection has proven remarkably effective, often achieving cure rates exceeding 90%, a testament to the microbiome's protective role. The urogenital tract employs different barrier strategies for its distinct anatomy and functions. The bladder and urethra are protected by the flushing action of urine. Regular urination mechanically removes bacteria before they can ascend and establish infection. Urine itself is typically sterile or contains very low bacterial counts and has mildly antimicrobial properties due to its composition, urea, uric acid, and varying pH depending on diet and hydration. The epithelium produces antimicrobial peptides and secretory IgA. The vagina maintains an acidic environment, typically pH 3.8 to 4.5, through the action of lactobacilli, beneficial bacteria that metabolize glycogen deposited in vaginal epithelial cells and produce lactic acid. This acidity inhibits most pathogenic bacteria and fungi. The lactobacilli also produce hydrogen peroxide and bacteriocins, providing additional antimicrobial protection. The vaginal microbiome's composition varies with menstrual cycle, pregnancy, and hormone levels, reflecting hormonal influences on epithelial glycogen deposition. The effectiveness of these barriers is perhaps best appreciated by considering what happens when they fail. Burn victims, having lost large areas of skin, face severe infection risk and often succumb to sepsis rather than the burn itself. Patients with cystic fibrosis, whose thickened mucus impairs mucociliary clearance, suffer chronic respiratory infections. Individuals with inflammatory bowel disease have compromised intestinal barrier function and increased bacterial translocation. Immunodeficiency disorders that affect antibody production often manifest as recurrent sinopulmonary infections, reflecting the importance of secretory IgA in mucosal protection. Yet even healthy barriers are not impenetrable. Pathogens have evolved numerous strategies to breach or circumvent these defenses. Some bacteria produce enzymes that degrade mucus, allowing them to reach the epithelium. Others possess adhesins, surface molecules that bind specifically to epithelial cell receptors, resisting mechanical clearance. Certain pathogens actively exploit barrier mechanisms. Poliovirus specifically targets and infects the M cells that sample intestinal contents for the immune system. Influenza virus specifically infects ciliated respiratory epithelial cells, disabling mucociliary clearance. Physical trauma obviously breaches skin barriers, which is why wound care emphasizes cleanliness and antiseptics. But more subtle breaches occur constantly. Minor abrasions from toothbrushing, microscopic tears in the intestinal epithelium, skin cracking in dry climates. Most of these go unnoticed because the immune system responds rapidly to contain any microbial invasion before it becomes clinically apparent. The transition from barrier breach to active infection depends on the virulence and number of invading pathogens, the integrity of local defenses, and the speed and effectiveness of the immune response. Understanding these barriers clarifies why hygiene, while important, can be taken to counterproductive extremes. The hygiene hypothesis proposes that excessive cleanliness and antimicrobial product use in developed countries reduces microbial exposure during childhood, impairing immune system development and contributing to allergies and autoimmune diseases. While controversial and likely oversimplified, this hypothesis highlights that some microbial exposure is necessary for proper immune education and that eliminating all microbes is neither possible nor desirable. The barriers we have examined work constantly and unconsciously, preventing the vast majority of potential infections before they begin. They are the immune system's first line of defense, unglamorous but essential. When barriers are breached and microbes enter the body's tissues, time becomes critical. A single bacterium can divide every 20 minutes under optimal conditions, producing millions of progeny within hours. The immune system cannot afford to wait for adaptive responses that require days to develop. Instead, innate cellular immunity responds within minutes to hours, deploying cells that can recognize and respond to broad categories of pathogens without prior exposure or specific training. These are the first responders, the phagocytes and natural killer cells that buy time for the more sophisticated adaptive responses to mobilize. The recognition problem is central to understanding innate immunity. How do these cells distinguish pathogens from the body's own tissues? The answer lies in pattern recognition receptors, proteins that detect molecular patterns shared by many microbes but absent from host cells. These pathogen associated molecular patterns, or PAMPs, include components of bacterial cell walls, viral nucleic acids, and fungal cell membranes, molecular signatures that betray an organism as foreign and potentially dangerous. Lipopolysaccharide, or LPS, a component of gram-negative bacterial outer membranes, is a prototypical PAMP. It is recognized by toll-like receptor 4, or TLR4, on macrophages, dendritic cells, and other immune cells. When TLR4 binds LPS, it triggers intracellular signaling cascades that activate the transcription factor NF-kappa B, which enters the nucleus and turns on genes for inflammatory cytokines, chemokines, and other immune response mediators. This is why even minute quantities of LPS, nanograms, can trigger inflammation. The recognition system is exquisitely sensitive. There are 10 functional toll-like receptors in humans, each recognizing different PAMPs. TLR2 recognizes components of gram-positive bacterial cell walls and mycobacterial cell envelopes. TLR3 detects double-stranded RNA, a signature of many viral infections. TLR5 recognizes flagellin, the protein that composes bacterial flagella. TLR7 and TLR8 detect single-stranded viral RNA. TLR9 recognizes unmethylated CpG DNA motifs, common in bacteria, but rare in mammalian DNA due to our extensive DNA methylation. These receptors are strategically distributed, some on cell surfaces to detect extracellular pathogens, others in endosomal compartments where they can detect nucleic acids from engulfed microbes or viruses replicating within cells. Beyond toll-like receptors, cells possess other pattern recognition receptor families. NOD-like receptors, or NLRs, reside in the cytoplasm and detect intracellular pathogens and danger signals. Some NLRs form multi-protein complexes called inflammasomes when activated, which activate caspase-1, which in turn cleaves pro-IL-1 beta into its active form. This mechanism ensures that the potent inflammatory cytokine IL-1 beta is only produced when genuine threats are detected. RIGI, like receptors, detect viral RNA in the cytoplasm and trigger interferon production. C-type lectin receptors on antigen-presenting cells recognize carbohydrate structures on pathogens. This multi-layered recognition system provides both sensitivity and specificity. Multiple different receptors must detect a pathogen before a full inflammatory response is triggered, reducing the chance of false alarms. Yet any one receptor can initiate defensive responses, ensuring that novel pathogens or those that evade one recognition mechanism may still be detected by others. The system also detects damage-associated molecular patterns, or DAMPs, molecules normally sequestered inside cells, but released when cells are damaged or die. These include heat shock proteins, high mobility group box 1 protein, and ATP. DAMPs alert the immune system to tissue damage, even in the absence of infection, initiating responses to promote repair and clear cellular debris. Neutrophils, as mentioned in Chapter 3, are the most abundant phagocytic cells and the first to arrive at sites of infection. Resident tissue macrophages, detecting PAMPs through their pattern recognition receptors, immediately begin secreting chemokines, particularly CXCL8, which creates a gradient attracting neutrophils from the bloodstream. Circulating neutrophils express receptors for CXCL8 and for other inflammatory mediators like leukotriene B4 and complement fragment C5a. When they encounter these signals, neutrophils slow their rapid circulation and begin rolling along the endothelial surface of blood vessels near the infection site. The endothelium itself has been activated by inflammatory cytokines like TNF alpha and IL-1 beta, increasing its expression of selectins, adhesion molecules that weakly bind to carbohydrates on the neutrophil surface. This weak binding causes neutrophils to roll along the vessel wall. If the chemokine gradient is sufficiently strong, indicating nearby infection, the neutrophil activates its integrins, much stronger adhesion molecules that firmly arrest the cell on the endothelium. The neutrophil then extends pseudopodia into the junction between endothelial cells and squeezes through, a process called transmigration or diaptesis, entering the tissue. Once in the tissue, neutrophils follow the chemokine gradient toward its source, moving with remarkable purpose and speed for a cell, up to 30 micrometers per minute. They can navigate complex, three-dimensional tissue environments, squeezing between collagen fibers and other cells. Upon reaching the infection site, they begin phagocytosis, the process of engulfing microbes. The neutrophil extends pseudopodia around a bacterium, surrounding it in a membrane-enclosed compartment called a phagosome. This phagosome then fuses with the neutrophil's granules, which are essentially specialized lysosomes, forming a phagolysosome. The phagolysosome is one of the most hostile environments in nature. The NADPH oxidase complex in the phagolysosomal membrane produces superoxide anions, which dismutate to form hydrogen peroxide. Myeloperoxidase, released from azurophilic granules, uses this hydrogen peroxide to produce hypochlorous acid, essentially bleach, one of the most potent antimicrobial agents known. Other reactive oxygen species, including hydroxyl radicals and singlet oxygen, are also generated. These oxidants damage essentially all biological molecules. They oxidize amino acids in proteins, peroxidize membrane lipids, and damage nucleic acids. The oxidative burst that produces these reactive species consumes enormous amounts of oxygen, sometimes so much that neutrophil-rich pus can be hypoxic despite adequate blood supply. This respiratory burst is not actual respiration, but rather the rapid consumption of oxygen for antimicrobial oxidant production. Individuals with chronic granulomatous disease, who have genetic defects in NADPH oxidase, cannot produce this respiratory burst. They suffer recurrent severe bacterial and fungal infections, particularly with catalase-positive organisms like Staphylococcus aureus and Aspergillus species, which destroy hydrogen peroxide and are thus especially resistant to other antimicrobial mechanisms. Beyond oxidative killing, the phagolysosome contains numerous antimicrobial proteins and peptides. Defensins insert into microbial membranes, creating pores that disrupt membrane integrity. Cathelicidin and other antimicrobial peptides have similar mechanisms. Proteolytic enzymes, including elastase, cathepsins, and metalloproteinases, degrade microbial proteins and help break down complex pathogens. Lactoferrin sequesters iron, starving bacteria of this essential nutrient. Lysozyme cleaves the peptidoglycan in bacterial cell walls, weakening them and making them more susceptible to osmotic lysis. Some bacteria have evolved resistance to various killing mechanisms, requiring neutrophils to employ multiple simultaneous attacks. Mycobacteria have thick, waxy cell walls, highly resistant to many antimicrobial agents. Some bacteria produce catalase or superoxide dismutase to neutralize reactive oxygen species. Others modify their surface structures to resist antimicrobial peptides. The continuous evolutionary arms race between pathogens and phagocytes has driven the development of multiple redundant killing mechanisms, ensuring that if one fails, others may succeed. Neutrophils can also release their antimicrobial arsenal extracellularly, particularly when faced with targets too large to phagocytose or when bacteria aggregate in biofilms. Frustrated phagocytosis occurs when neutrophils repeatedly attempt to engulf something unsuccessfully, ultimately degranulating and releasing their contents extracellularly. This causes collateral damage to surrounding tissues, a significant source of pathology in chronic inflammatory conditions. More dramatically, neutrophils can undergo endotosis, expelling their DNA decorated with histones and granular proteins to form neutrophil extracellular traps, or NETs. Macrophages are present in all tissues as long-lived residents. These tissue macrophages have different names in different locations: alveolar macrophages in lung, Kupffer cells in liver, microglia in brain, but all serve similar functions: phagocytosis of pathogens and debris, antigen presentation, and secretion of cytokines that shape immune responses. During infection, circulating monocytes are recruited from the bloodstream and differentiate into additional macrophages, supplementing the resident population. Macrophages phagocytose bacteria using similar mechanisms as neutrophils, but are generally longer lived and can continue killing for days to weeks. They express numerous receptors that recognize pathogens. Toll-like receptors detect PAMPs, complement receptors bind C3b-coated bacteria, FC receptors bind antibody-coated pathogens, and scavenger receptors recognize modified host molecules and some bacterial components. Plasmacytoid dendritic cells, a specialized subset, are particularly important in antiviral immunity. They detect viral nucleic acids through TLR7 and TLR9 in endosomes and respond by producing enormous quantities of type I interferons, up to 1,000-fold more than other cell types. These interferons create an antiviral state in surrounding cells, limiting viral spread during the critical first days of infection before adaptive immunity can be mobilized. Plasmacytoid dendritic cells are found in blood and lymphoid tissues, positioned to detect blood-borne viruses and viruses that have reached lymphoid organs. Natural killer cells, or NK cells, provide innate defense against virus-infected and cancerous cells. As discussed in Chapter 3, they recognize targets through a balance of activating and inhibitory signals. Their inhibitory receptors bind MHC class I, which is expressed on all healthy nucleated cells. Many viruses downregulate MHC class I expression to evade CD8+ T cells, but this makes infected cells vulnerable to NK cells, which lose their inhibitory signal and attack. Tumor cells sometimes also lose MHC class I expression during malignant transformation, again rendering them susceptible to NK cells. NK cells also recognize stress induced molecules on infected or transformed cells through activating receptors like NKG2D. When activating signals predominate over inhibitory signals, the NK cell forms a cytotoxic synapse with the target, releases granules containing perforin and granzymes, and induces target cell apoptosis. NK cells also produce IFN gamma, which activates macrophages and influences adaptive immunity toward Th1 type responses. Early in viral infections, before adaptive immunity develops, NK cells provide crucial defense, and individuals with NK cell deficiencies suffer severe herpesvirus infections. The coordination among these innate immune cells is remarkable. Resident macrophages detect pathogens and immediately secrete cytokines and chemokines, alerting the immune system and recruiting neutrophils. Neutrophils arrive within hours and begin killing bacteria, though causing some collateral tissue damage. Monocytes arrive and differentiate into macrophages that help with pathogen clearance, phagocytose dead neutrophils, and orchestrate healing. Dendritic cells capture antigens and migrate to lymph nodes to activate adaptive immunity. NK cells eliminate virus-infected cells. All of these processes occur in parallel, creating a coordinated response that is greater than the sum of its parts. This cellular innate response buys time for adaptive immunity, but also shapes what adaptive responses will develop. The cytokines secreted by innate immune cells, the antigens captured by dendritic cells, and the level of inflammation all influence how T and B cells respond. A bacterial infection inducing IL-12 production will generate Th1 responses and antibodies that enhance phagocytosis. A parasitic infection generating IL-4 will produce T responses and IgE antibodies that activate eosinophils. The innate response provides the instructions that tell adaptive immunity what type of response is needed. Yet innate cellular immunity alone is often insufficient. While it can clear many bacterial infections and contain viral infections temporarily, complete elimination of intracellular pathogens and development of long-lasting immunity requires adaptive responses. And the innate response itself, while protective, also causes inflammation, with symptoms that are often the primary burden of infectious disease. The fever, pain, swelling, and malaise of infection are largely due to innate immune responses, particularly the cytokines produced by macrophages and the tissue damage caused by neutrophils. This collateral damage is an acceptable cost during acute infection, but becomes problematic when inflammation is prolonged. Chronic inflammation, whether due to persistent infection, autoimmunity, or other causes, is implicated in numerous diseases, including atherosclerosis, diabetes, cancer, and neurodegenerative disorders. The transition from innate to adaptive immunity requires specific molecular events, particularly the process of antigen presentation. This is the subject of our next chapter, where we will examine how proteins from pathogens are processed, displayed on MHC molecules, and presented to T cells. The critical link between innate detection and adaptive response. Inflammation is perhaps the most familiar manifestation of the immune system at work. The cardinal signs rubor, tumor, calor, dolor, and functio laesa Redness, swelling, heat, pain, and loss of function were described by ancient physicians and remain relevant today. These signs reflect the local vascular and cellular changes that occur when the immune system responds to infection, injury, or other threats. Inflammation is fundamentally protective, recruiting immune cells and delivering antimicrobial substances to sites where they are needed. Yet it is also a source of considerable discomfort and, when excessive or prolonged, a cause of tissue damage and disease. The inflammatory cascade begins with recognition of danger. This might be PAMPs, detected by tissue-resident macrophages encountering bacteria, DAMPs released from damaged cells after injury, or allergens cross-linking IgE on mast cells. Regardless of the trigger, the immediate result is similar. Cells at the site begin secreting inflammatory mediators, small molecules that alter vascular function and recruit immune cells. These mediators include preformed substances released from granules, like histamine from mast cells, and newly synthesized compounds like prostaglandins, leukotrienes, and cytokines. Histamine, stored in basophils and mast cells and rapidly released upon activation, is one of the first mediators to act. It binds to histamine receptors on endothelial cells lining nearby blood vessels, causing them to contract slightly. This contraction creates gaps between endothelial cells, increasing vascular permeability. Fluid leaks from the bloodstream into surrounding tissues, causing swelling or edema. This fluid contains proteins, including antibodies and complement factors, which contribute to pathogen elimination. Histamine also causes vasodilation, the widening of blood vessels, which increases blood flow to the area, producing the redness and heat characteristic of inflamed tissue. The increased blood flow serves multiple functions. It delivers more oxygen and nutrients to support the increased metabolic activity of immune responses. It raises local temperature, which can inhibit some temperature-sensitive pathogens and accelerate enzymatic reactions. It brings more immune cells circulating past the inflamed site, increasing the chance they will be recruited. The warmth you feel when touching inflamed skin reflects this enhanced perfusion, the tissue literally running hotter than surrounding areas. Prostaglandins and leukotrienes are lipid mediators synthesized from arachidonic acid, a polyunsaturated fatty acid released from cell membranes by phospholipase A2. Two enzymatic pathways process arachidonic acid. Cyclooxygenase produces prostaglandins and thromboxanes, while lipoxygenase produces leukotrienes. Different prostaglandins have distinct effects. Prostaglandin E2, or PGE2, promotes vasodilation, increases vascular permeability, and sensitizes nerve endings to pain. It also acts on the hypothalamus to raise the body's temperature set point, producing fever. Prostaglandin 1,2, or prostacyclin, also promotes vasodilation and inhibits platelet aggregation, preventing unwanted clotting. Nonsteroidal anti-inflammatory drugs, or NSAIDs, including aspirin, ibuprofen, and naproxen, work by inhibiting cyclooxygenase enzymes, reducing prostaglandin production. This explains their therapeutic effects, reducing inflammation, lowering fever, and relieving pain. It also explains some of their side effects. Prostaglandins in the stomach promote mucus secretion that protects the gastric lining, so inhibiting prostaglandin production can cause gastric ulcers. Prostaglandins in the kidney regulate blood flow and fluid balance, so NSAID use can impair kidney function, particularly in vulnerable individuals. Leukotrienes, Products of the lipoxygenase pathway are potent inflammatory mediators. Leukotriene, B4, is a powerful chemoattractant for neutrophils, guiding them from the bloodstream to infection sites. Cysteine leukotrienes, including LTC4, LTD4, and LTE4, cause bronchoconstriction, increase vascular permeability, and promote mucus secretion. Cytokines amplify and propagate inflammatory responses. TNF alpha and IL-1 beta, secreted by activated macrophages and other cells, have numerous pro-inflammatory effects. They activate endothelial cells, increasing expression of adhesion molecules like E-selectin, P-selectin, and ICAM, one that capture passing leukocytes. They stimulate fibroblasts and epithelial cells to secrete chemokines that guide leukocyte migration. They promote synthesis of acute phase proteins in the liver, including C-reactive protein and serum amyloid A, which opsonize bacteria and modulate inflammation. They act on the hypothalamus to induce fever and on muscle and fat tissue to promote catabolism, mobilizing energy stores to fuel immune responses. IL-6, another important inflammatory cytokine, serves as a bridge between innate and adaptive immunity. It promotes acute phase protein synthesis, induces fever, and stimulates B cell maturation into antibody-secreting plasma cells. IL-6 also promotes differentiation of CD4 plus T cells into Th17 cells, which produce IL-17 and promote neutrophil recruitment. Elevated IL-6 levels are associated with many inflammatory diseases, and IL-6 blockade with therapeutic antibodies has proven effective for treating rheumatoid arthritis, among other conditions. Chemokines create the molecular GPS that guides leukocyte migration. During inflammation, tissue cells and resident immune cells secrete chemokines that diffuse through tissues and establish concentration gradients. Leukocytes expressing appropriate chemokine receptors detect these gradients and migrate toward higher concentrations, a process called chemotaxis. Different chemokines attract different cell types, allowing selective recruitment. CXCL8 attracts neutrophils during bacterial infection. CCL2 recruits monocytes. CCL5 brings in T cells. CCL11 recruits eosinophils during parasitic infection or allergic inflammation. The complement system, a cascade of plasma proteins, provides another layer of inflammatory amplification. Complement activation can occur through three pathways. The classical pathway triggered by antibody-antigen complexes, the lectin pathway triggered by mannose-binding lectin recognizing bacterial carbohydrates, and the alternative pathway triggered by bacterial surface structures. All three converge on the formation of C3 convertase enzymes that cleave C3, the central component, into C3A and C3B. C3A is an anaphylatoxin, a small fragment that binds to receptors on mast cells and basophils, causing degranulation and histamine release. It also acts on smooth muscle, causing contraction, and recruits neutrophils and monocytes. C3b binds covalently to pathogen surfaces, acting as an opsonin that greatly enhances phagocytosis. Phagocytes express complement receptors that bind C3b-coated particles, and this binding triggers much more efficient uptake than recognition of uncoated pathogens. The complement cascade continues beyond C3, generating C5 convertases that cleave C5 into C5a and C5b. C5a is an even more potent anaphylatoxin than C3A and a powerful chemoattractant for neutrophils. C5b initiates the terminal complement cascade, recruiting C6, C7, C8, and multiple copies of C9 to form the membrane attack complex, or MAC. This complex inserts into cell membranes, creating pores that disrupt osmotic balance and can directly lyse bacteria. The MAC is particularly effective against Neisseria bacteria, which cause meningitis and gonorrhea. Individuals with deficiencies in terminal complement components suffer recurrent Neisserial infections. The vascular changes during inflammation serve not only to deliver immune effectors, but also to contain infection. The leaked fluid dilutes toxins and carries antimicrobial substances. Fibrinogen, leaking from blood into tissues, is converted to fibrin, creating a meshwork that walls off the infected area and limits pathogen spread. This containment is a double-edged sword. It prevents systemic dissemination of infection, but can also impair tissue function and, if excessive, cause thrombosis. The balance between containing infection and maintaining blood flow is delicate and sometimes goes awry, particularly in sepsis, where widespread coagulation causes disseminated intravascular coagulation, or DIC. Pain, one of the cardinal signs of inflammation, serves an important protective function despite its unpleasantness. Inflammatory mediators, particularly prostaglandins, bradykinin, and substance P, sensitize nociceptors, the nerve endings that detect painful stimuli. This sensitization lowers the threshold for pain, so that even light touch to inflamed tissue becomes painful. The pain promotes protective behavior, encouraging rest and preventing further tissue damage. An inflamed joint that hurts with movement will be rested, allowing healing. However, chronic inflammatory pain serves no useful purpose and becomes a source of suffering, contributing to the burden of chronic inflammatory diseases. The resolution of acute inflammation is an active process, not simply the passive dissipation of inflammatory stimuli. As pathogens are cleared and damaged tissue is removed, the inflammatory program must be terminated and healing must begin. This involves a shift from pro-inflammatory to anti-inflammatory mediators, a process coordinated by multiple mechanisms. Macrophages that have phagocytosed apoptotic neutrophils, a sign that the battle is won, switch their cytokine profile from pro-inflammatory TNF-alpha, IL-1 beta, to anti-inflammatory IL-10, TGF-beta. Lipoxins are produced from arachidonic acid through sequential actions of different lipoxygenases. Aspirin, interestingly, promotes lipoxin production by acetylating COX-2, which then produces 15-R-HEDE, a precursor for aspirin-triggered lipoxins. This may contribute to aspirin's anti-inflammatory effects beyond simple prostaglandin inhibition. Resolvins and protectins are produced from omega-3 fatty acids, EPA and DHA, explaining some of the anti-inflammatory benefits attributed to these dietary fatty acids. Macrophages recognize phosphatidylserine exposed on the surface of apoptotic cells and engulf them. This engulfment triggers anti-inflammatory signaling in the macrophage, creating a feedback loop that promotes resolution. Defects in efferocytosis are implicated in chronic inflammatory and autoimmune diseases, where accumulation of apoptotic cell debris can provide autoantigens that trigger autoimmunity. When inflammation fails to resolve, chronic inflammation ensues. This can occur for several reasons: persistent infection with pathogens that resist clearance, continued exposure to inflammatory stimuli like inhaled pollutants or allergens. Autoimmunity, where the immune system attacks self tissues, or defects in resolution mechanisms. Chronic inflammation is characterized by a different cellular infiltrate than acute inflammation. Rather than neutrophils, chronic inflammatory lesions contain macrophages, lymphocytes, and plasma cells. The tissue architecture becomes disrupted, with fibrosis replacing normal structures and function progressively deteriorating. Many common diseases involve chronic inflammation. Atherosclerosis, the buildup of plaques in arterial walls, involves chronic inflammation driven by modified lipoproteins that activate macrophages. These macrophages accumulate lipid, becoming foam cells, and secrete inflammatory mediators that promote plaque growth. Eventually, plaques can rupture, triggering thrombosis and causing heart attacks or strokes. Type 2 diabetes involves chronic low-grade inflammation, with inflammatory cytokines interfering with insulin signaling. Obesity itself is associated with increased inflammation, as adipose tissue secretes inflammatory mediators. Chronic inflammatory diseases are often treated with immunosuppressive or anti-inflammatory drugs. Corticosteroids, synthetic analogs of cortisol, are potent anti-inflammatory agents that suppress multiple aspects of inflammation. They inhibit phospholipase, A2, reducing prostaglandin and leukotriene production. They inhibit NF-kappa B, reducing cytokine gene expression. They promote neutrophil apoptosis and inhibit macrophage activation. However, their broad suppression of immunity increases infection risk, and long-term use causes numerous side effects, including bone loss, muscle wasting, and metabolic disturbances. Disease-modifying antihumatic drugs, or DMARDs, target specific components of inflammatory pathways. Methotrexate inhibits folate metabolism and has anti-inflammatory effects through multiple mechanisms. Sulfasalazine has anti-inflammatory and immunomodulatory properties. Hydroxychloroquine modulates antigen presentation and inflammatory signaling. These drugs are less potently immunosuppressive than corticosteroids, but still require monitoring for adverse effects. Biologic therapies targeting specific cytokines or cell surface molecules have revolutionized treatment of inflammatory diseases. TNF inhibitors, IL-6 inhibitors, IL-17 inhibitors, and inhibitors of other cytokines provide targeted suppression of inflammation with more favorable safety profiles than broadly immunosuppressive drugs, though they still increase infection risk. Antibodies targeting T cell or B cell surface molecules can deplete these cells or block their activation, useful in autoimmune diseases. The development of these targeted therapies has vindicated decades of basic research into inflammatory mechanisms, demonstrating that understanding immunity at a molecular level translates into better treatments. Inflammation, then, is neither inherently good nor bad, but rather a powerful tool that must be appropriately deployed and controlled. Acute inflammation clears infections and promotes healing. Chronic inflammation destroys tissues and causes disease. The transition from acute to chronic inflammation and the failure to resolve inflammation properly represents a critical juncture in many diseases. Understanding inflammation at molecular, cellular, and systemic levels is essential for developing better anti-inflammatory therapies that preserve protective immunity while preventing pathological inflammation. The inflammatory response is intimately connected to antigen presentation, the process by which professional antigen-presenting cells capture antigens at inflammatory sites and present them to T cells in lymph nodes. initiating adaptive immunity. It is to this crucial bridge between innate and adaptive immunity that we now turn. The thymus is where T cells learn to distinguish self from non self, a process so rigorous that only about 2% of developing T cells survive to maturity. This selection process is necessary because T cell receptors are generated through random genetic recombination, a mechanism that creates enormous diversity but also produces many receptors that would be useless or actively harmful. Some T cell receptors cannot recognize MHC molecules at all and thus could never be activated. Others recognize self-antigens too strongly and would cause autoimmune disease if allowed to mature. The thymus eliminates both types, ensuring that only functional, safe T cells enter circulation. The journey begins when T cell progenitors leave the bone marrow and migrate to the thymus, entering through blood vessels in the corticomedullary junction. These cells, called thymocytes in their immature thymic stage, are double negative, expressing neither CD4 nor CD8. They begin proliferating and rearranging their T cell receptor genes, a process involving VDJ recombination, similar to that in B cells but involving different gene segments. The T cell receptor beta chain genes are rearranged first, combining variable V, diversity D, and joining J segments to create a unique beta chain sequence. If beta chain rearrangement is successful, the beta chain pairs with a pre-T alpha chain to form the pre-T cell receptor, which signals the cell to stop rearranging the beta chain locus, ensuring each T cell expresses only one beta chain, a process called allelic exclusion. The cell also begins expressing both CD4 and CD8, becoming double positive, and starts rearranging the alpha chain genes. Multiple rounds of alpha chain rearrangement can occur, giving more chances for success until a functional T cell receptor alpha chain is produced that pairs with the beta chain to form a complete T cell receptor. At this point, the double positive thymocyte has a functional T cell receptor but hasn't yet been tested. The cell is in the thymic cortex, surrounded by cortical epithelial cells that express both MHC class I and MHC class II, loaded with various self-peptides. The thymocyte's T cell receptor engages these MHC peptide complexes, and the strength of this engagement determines the cell's fate. Positive selection occurs first. Thymocytes, whose T cell receptors can bind to MHC molecules, either class I or class II, with sufficient affinity, receive survival signals. Those whose receptors cannot bind MHC at all, or bind too weakly, receive no signal and die by neglect, undergoing apoptosis within 3 to 4 days. This eliminates T cells that would be unable to function in the periphery, where T cell activation requires T cell receptor engagement with MHC peptide complexes. Positive selection also determines CD4 versus CD8 lineage commitment. Cells whose receptors bind MHC class II receive signals that suppress CD8 expression and promote CD4 expression, becoming CD4 plus single positive cells, while those whose receptors bind MHC class I suppress CD4 and maintain CD8 expression. The molecular mechanisms of positive selection are complex. Engagement of the T cell receptor with MHC peptide delivers signals through the associated CD3 complex and activates intracellular signaling cascades involving kinases like LUC and ZAP70. The strength and duration of signaling, influenced by the affinity of the interaction and the amount of the selecting ligand, determine the outcome. Intermediate signaling strength promotes survival and differentiation, while very weak signaling leads to death by neglect. Surviving the cortex, now single positive for either CD4 or CD8, thymocytes migrate to the thymic medulla. Here they encounter a different environment and a second, more stringent test: negative selection. The medulla contains dendritic cells and medullary thymic epithelial cells that express a remarkable feature. They display not just the limited set of proteins normally expressed in the thymus, but also tissue-specific antigens from throughout the body. This ectopic expression is controlled by the AIR gene autoimmune regulator, which allows medullary epithelial cells to express and present insulin from the pancreas, thyroglobulin from the thyroid, myelin proteins from the nervous system, and hundreds of other tissue-specific proteins. This mechanism allows developing T cells to be exposed to virtually the entire repertoire of self-antigens before leaving the thymus. Any thymocyte whose T cell receptor binds too strongly to these self-peptide MHC complexes receives a death signal and undergoes apoptosis. This is negative selection, the elimination of potentially autoreactive T cells. The threshold for negative selection is higher than for positive selection. The TCR-MHC interaction must be quite strong to trigger deletion. This creates a window of intermediate affinity where T cells survive both selection steps, strong enough binding for positive selection, but not so strong as to trigger negative selection. The distinction between positive and negative selection is not absolute, but reflects differences in signaling strength and context. In simplified terms, weak TCR engagement in the cortex promotes survival. Strong engagement in the medulla promotes death. In reality, the duration of signaling, the integration of multiple signals over time, the developmental stage of the thymocyte, and the presence of other co-stimulatory or co-inhibitory signals all influence the outcome. Some thymocytes that recognize self-antigens with high affinity do not undergo apoptosis, but instead differentiate into regulatory T cells, or TREGs. These cells express the transcription factor FOXP3 and are specialized for suppressing immune responses and maintaining tolerance. Thymic TREGs, or T-TREGs, constitute about 5 to 10% of CD4 plus T cells leaving the thymus and play crucial roles in preventing autoimmunity. The signals that divert high-affinity, self-reactive thymocytes into the TREG lineage rather than deletion are incompletely understood, but involve strong TCR signaling combined with particular cytokine signals, especially IL-2. Mutations in ARR cause a rare autoimmune syndrome called autoimmune polyendocrinopathy, candidiasis ectodermal dystrophy, or APECD. Patients with this condition develop autoimmune attacks against multiple endocrine organs because their medullary epithelial cells cannot express tissue-specific antigens. Autoreactive T cells, specific for pancreatic, thyroid, adrenal, and other antigens, escape negative selection and attack these tissues in the periphery. This demonstrates the critical importance of central tolerance, the tolerance established in the thymus, for preventing autoimmunity. Not all self-reactive T cells are deleted in the thymus. Some self-antigens are not expressed at sufficient levels in the thymus to trigger negative selection. Some T cells have intermediate affinity for self-antigens and pass through thymic selection, but could still be activated under the right conditions in the periphery. This is where peripheral tolerance mechanisms become important. Tissues maintain tolerance through several mechanisms: absence of co-stimulatory molecules, so autoreactive T cells receive signal 1 without signal 2 and become anergic. Expression of inhibitory molecules like PDL, one that deliver inhibitory signals to T cells. Secretion of anti-inflammatory cytokines like TGF-beta and IL-10, and the presence of peripheral regulatory T cells that actively suppress autoreactive responses. The thymus itself changes dramatically with age in a process called thymic involution. The thymus is largest relative to body size in infancy and early childhood, then gradually shrinks, with thymic tissue increasingly replaced by adipose tissue. By adulthood, the thymus may be less than 10% its peak size, though it retains some function throughout life. This involution reduces the production of new naive T cells, and the peripheral T cell pool becomes increasingly dominated by memory T cells generated earlier in life. Thymic involution contributes to immunosenescence, the age-related decline in immune function. Older individuals have reduced ability to respond to novel pathogens or vaccines because they generate fewer new T cells with diverse specificities. The T cell repertoire becomes increasingly restricted, focused on antigens encountered earlier in life. There is increased susceptibility to new infections and reduced vaccine efficacy. Strategies to reverse or slow thymic involution are being investigated, including growth hormone treatment, sex steroid ablation, and regenerative approaches, but remain experimental. The enormous loss of cells during thymic selection, with 98% dying, seems wasteful but is necessary given how T cell receptors are generated. The random recombination that creates TCR diversity cannot be constrained to produce only useful, non-autoreactive receptors. Instead, the thymus generates vast numbers of T cells with random specificities and then selects the useful ones. It is quality control through mass production and rigorous testing. The selection process also explains MHC restriction. T cells are positively selected on the MHC molecules expressed by thymic epithelial cells, which are the individual's own MHC molecules. Thus, mature T cells can only function with these MHC molecules. They cannot recognize peptides presented by different MHC molecules, even if the peptide is identical. This creates a fundamental compatibility requirement for T cell responses and explains why transplanted organs expressing foreign MHC molecules are recognized and rejected. Some pathogens exploit thymic selection. HIV infects and destroys CD4 plus T cells, progressively depleting them and leading to immunodeficiency. But HIV also infects thymic epithelial cells and thymocytes, impairing thymic function and reducing the generation of new T cells. This exacerbates the CD4 plus T cell loss caused by peripheral infection. The thymus attempts to compensate by increasing activity, but in chronic HIV infection, thymic function progressively deteriorates, contributing to the profound immunodeficiency of AIDS. The education of T cells in the thymus represents one of the most sophisticated quality control systems in biology. It solves the fundamental problem of generating useful immune diversity while avoiding autoimmunity. It creates T cells that can recognize foreign antigens presented on the body's own MHC molecules while remaining tolerant to self. It establishes a diverse repertoire capable of responding to the unpredictable universe of pathogens, and it generates regulatory T cells that provide an additional layer of self-tolerance and immune regulation. The T cells that emerge from the thymus are educated but naive, having never encountered their specific antigen in the periphery. They are like graduates, equipped with training, but no real world experience. They circulate through blood and lymphoid tissues, surveying antigens presented by professional antigen presenting cells, waiting for the encounter that will activate them and allow them to perform their functions. When that encounter occurs, these naive T cells transform into effector cells capable of orchestrating or directly executing immune responses. It is to these cell mediated immune responses that we now turn. Naive T cells exit the thymus and enter the bloodstream, joining the continuous circulation of lymphocytes through blood, tissues, and lymphoid organs. These cells are mature but inexperienced, carrying T cell receptors specific for particular peptide MHC combinations, but having never encountered those antigens. They are small quiescent cells with minimal cytoplasm and little metabolic activity, essentially waiting for activation. Most will circulate for weeks or months without finding their specific antigen, continuously surveying but finding nothing to respond to. But when a naive T cell's receptor recognizes its cognate antigen presented under the right conditions, everything changes. T cell activation typically occurs in secondary lymphoid organs, particularly lymph nodes. Naive T cells enter lymph nodes from the blood through specialized postcapillary venules called high endothelial venules, or HEVs. These vessels have tall endothelial cells that express addresins, which bind to L-selectin on naive T cells, allowing the T cells to extravasate into the lymph node parenchyma. Once inside, T cells migrate through the paracortex, the T cell zone, where they briefly contact numerous dendritic cells, surveying the MHC peptide complexes each dendritic cell displays. Most T cells find no relevant antigen and exit via the efferent lymphatic, eventually returning to blood and continuing their circulation. But if a T cell encounters a dendritic cell displaying its specific antigen, the T cell stops migrating, forms a stable interaction with the dendritic cell, and begins the activation process. This stable interaction can last hours to days, with the T cell and dendritic cell forming an immunological synapse, a specialized contact zone where signaling molecules cluster. The immunological synapse has a characteristic structure, sometimes called a supramolecular activation cluster, or SMAC. At the center is a cluster of T cell receptors engaging MHC peptide complexes, along with coreceptors CD4 or CD8 and signaling molecules. Surrounding this central region is a peripheral zone containing adhesion molecules like LFA1 and ICAM1 that strengthen the interaction. This organization facilitates sustained signaling and prevents premature disengagement. For full activation, the naive T cell requires 3 signals. Signal 1 is T cell receptor engagement by specific peptide MHC complexes. Signal 2 is co-stimulation, typically B7 molecules on the dendritic cell binding to CD28 on the T cell. Signal 3 is cytokines that influence differentiation. If all 3 signals are provided, the T cell undergoes a remarkable transformation. It enlarges, developing abundant cytoplasm and organelles. It begins proliferating rapidly, dividing every 4 to 6 hours for several days, generating a large clone of antigen-specific T cells, and it differentiates into effector cells capable of performing immune functions. The proliferative response is massive. A single activated T cell can generate thousands of progeny within a week. This clonal expansion is necessary because naive T cells specific for any given antigen are extremely rare, perhaps 1 in 100,000 to 1 in a million T cells. To mount an effective response, this tiny population must expand substantially. At the peak of the response, antigen-specific T cells may constitute 10 to 30% of all T cells in a lymph node or spleen, a 10,000-fold or greater increase from baseline. CD8 plus T cells that recognize their antigen presented on MHC class I, typically by dendritic cells that have cross-presented antigens from virus-infected cells or tumors, differentiate into cytotoxic T lymphocytes or CTLs. These effector CD8 plus T cells express high levels of cytotoxic molecules, including perforin, granzymes, and Fas ligand, and they home to sites of infection or tumor location. CTLs recognize infected or abnormal cells through the same MHC class I peptide complexes that activated them in the lymph node, but now they kill rather than merely recognize. The killing mechanism involves formation of a cytotoxic synapse between the CTL and its target. The CTL reorients its cytoskeleton and organelles, directing its cytotoxic granules toward the synapse. These granules fuse with the plasma membrane and release their contents in a directed fashion toward the target cell, minimizing bystander damage. Perforin polymerizes in the target cell membrane, forming pores. Granzymes, serine proteases, enter through these pores and activate caspases, proteases that execute the apoptotic program. The target cell undergoes apoptosis, dying in a controlled fashion. Its nuclear DNA is fragmented, its mitochondria release cytochrome C, and its plasma membrane develops blebs, but the cell remains intact until phagocytes remove it. This is crucial. If infected cells underwent necrosis, releasing their contents, viruses could escape and infect nearby cells. Apoptosis eliminates the cell without releasing infectious particles. The CTL detaches after delivering the lethal hit and can go on to kill additional targets. A single CTL can kill multiple infected cells sequentially, each killing taking just a few minutes. CTLs also kill through Fas ligand interactions. Fas ligand on the CTL binds to Fas, also called CD95, on the target cell. This binding causes Fas molecules to trimerize and recruit adapter proteins that activate caspase 8, initiating the extrinsic apoptotic pathway. This mechanism is particularly important for killing cells that have acquired resistance to perforin granzyme-mediated death. The two pathways provide redundancy, ensuring that targets can be eliminated even if one pathway is defective or inhibited. CD4 plus T cells, recognizing their antigen presented on MHC class II by professional antigen-presenting cells, differentiate into helper T cells with several possible phenotypes. The differentiation pathway depends largely on signal 3, the cytokine environment during activation. IL-12, produced by dendritic cells and macrophages responding to intracellular bacteria or viruses, drives differentiation into Th1 cells. IL-4, whose initial sources are somewhat unclear but include NKT cells and perhaps basophils, promotes Th2 differentiation, TGF-beta, plus IL-6, induce Th17 cells. TGF-beta alone can promote regulatory T-cell differentiation. Th1 cells are characterized by production of IFN-gamma and TNF-alpha. They activate macrophages, enhancing their microbicidal activity, particularly against intracellular pathogens like mycobacteria and listeria. IFN-gamma from Th1 cells activates macrophages to produce more nitric oxide and reactive oxygen species, increasing killing of ingested microbes. Th1 cells also promote B cell class, switching to IgG subclasses that are effective opsonins and complement activators, enhancing phagocytosis of extracellular bacteria. The Th1 response is crucial for clearing intracellular infections. Mice or humans with defects in IL-12, IFN gamma, or their receptors suffer severe mycobacterial infections, including infections with normally benign environmental mycobacteria. The chronic inflammation caused by unchecked intracellular bacteria leads to granuloma formation, organized aggregates of macrophages and T cells attempting to contain infection. While granulomas can sequester bacteria and prevent dissemination, they also cause tissue damage, the pathological basis of tuberculosis. Two cells produce IL-4, IL-5, and IL-13. They promote B cell class switching to IgE, recruit and activate eosinophils, and stimulate alternatively activated M2 macrophages. This response pattern is effective against helminthic parasites. IgE antibodies coat the worms, and eosinophils recognize the antibody-coated parasites through Fc receptors, attaching and degranulating to release toxic proteins onto the worm's surface. Two responses also promote tissue repair after injury, with M2 macrophages producing growth factors and extracellular matrix components. However, two responses are also responsible for allergic diseases. The same IgE meant to combat parasites can be produced against harmless environmental antigens like pollen or food proteins. Mast cells and basophils bearing IgE receptors degranulate when allergens cross-link bound IgE, causing allergic symptoms from hay fever to anaphylaxis. The hygiene hypothesis suggests that reduced parasite exposure in developed countries leads to inappropriate Th2 responses against harmless antigens, contributing to the allergy epidemic, though this remains debated. Th17 cells secrete IL-17A, IL-17F, and IL-22. They recruit neutrophils and promote antimicrobial peptide production by epithelial cells, important for defense against extracellular bacteria and fungi, particularly at mucosal surfaces. The 17 responses protect against Candida albicans infections. Individuals with defects in IL-17 signaling suffer chronic mucocutaneous candidiasis. However, excessive Th17 responses contribute to autoimmune diseases, including psoriasis, inflammatory bowel disease, and rheumatoid arthritis. The balance between protective and pathological Th17 activity is delicate. Regulatory T cells, or TREGs, produce IL-10 and TGF-beta and suppress immune responses. They can arise in the thymus as mentioned in the previous chapter, or differentiate from naive CD4 plus T cells in the periphery under certain conditions, these being called peripheral TREGs or PTREGs. TREGs express FOXP3, a transcription factor essential for their suppressive function. They suppress through multiple mechanisms, secreting anti-inflammatory cytokines, consuming IL-2, thus depriving effector T cells of this growth factor, expressing CTLA-4, which outcompetes CD28 for B7 binding and delivers inhibitory signals and direct killing of effector T cells or antigen-presenting cells. TREGs are essential for preventing autoimmunity and limiting excessive inflammation. Humans or mice lacking functional FOXP3 develop severe fatal autoimmune disease affecting multiple organs. IPEX syndrome, immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome results from FOXP3 mutations and causes autoimmune diabetes, thyroiditis, enteropathy, and other manifestations, often fatal in infancy without treatment. This demonstrates that active suppression, not just absence of autoreactive cells, is necessary to maintain tolerance. Follicular helper T cells, or TFH cells, represent another CD4 plus T cell subset specialized for helping B cells. These cells express CXCR5, allowing them to migrate into B cell follicles and produce IL-21, which promotes B cell proliferation and differentiation. TFH cells are essential for germinal center reactions, the sites where B cells undergo affinity maturation and class switching, processes we will examine in the next chapter. They select high-affinity B cells for survival while low-affinity B cells die, optimizing antibody quality. The different helper T cell subsets exhibit mutual antagonism. IFN gamma from Th. One cells inhibits Th2 differentiation. IL-4 from Th2 cells inhibits Th1 differentiation. This creates a tendency toward polarized responses. Infections typically elicit predominantly Th1 or Th2 responses rather than balanced combinations. This polarization can be disadvantageous, inappropriate. Two responses to intracellular bacteria worsen disease, and excessive Th1 responses can cause immunopathology. But polarization may also prevent conflicting responses that would interfere with each other. Memory T cells represent the long-term outcome of T cell activation. Most activated T cells die after the pathogen is cleared in a process called contraction. The massive clonal expansion is followed by apoptosis of 90 to 95% of effector cells once antigen and inflammation subside, but 5 to 10% survive and persist as memory T cells. These cells are long-lived, surviving years to decades, and can respond much more rapidly than naive T cells upon re-encounter with their antigen. Memory T cells are heterogeneous. Central memory T cells, or TCM cells, express lymph node homing receptors and reside primarily in lymphoid tissues. Upon reactivation, they proliferate extensively and generate effector cells. Effector memory T cells, or TEM cells, express tissue homing receptors and circulate through peripheral tissues. They can perform effector functions immediately upon antigen encounter without needing to proliferate first. Tissue-resident memory T cells, or TRM cells, lodge in peripheral tissues like skin, lung, and gut, and do not circulate. They provide immediate protection at sites of previous infection. The distribution of memory T cells explains why immunity to some pathogens is long-lasting while immunity to others wanes. Infections that generate robust memory T cell responses, particularly TRM cells, at sites of pathogen entry, provide durable protection. Respiratory viruses like influenza generate lung TRM cells that protect against reinfection. Skin infections generate skin TRM cells. Infections that generate primarily circulating memory cells may provide less immediate protection, as memory cells must be recruited to infection sites and may arrive too late to prevent illness. T cell exhaustion represents a distinct state that occurs during chronic infections or cancer. When T cells experience prolonged antigen exposure and inflammation, they progressively lose effector functions, a process mediated by increased expression of inhibitory receptors like PD-1, CTLA-4, LAG3, and TIM3. Exhausted T cells have reduced proliferative capacity, diminished cytokine production, and impaired cytotoxicity. This prevents immunopathology during chronic infections but allows pathogens or tumors to persist. Checkpoint inhibitor therapies that block PD-1 or CTLA-4 can reverse exhaustion and restore T cell function, which is why they are effective in some cancers. However, reversing exhaustion also risks immunopathology and autoimmunity, which explains the autoimmune side effects termed immune-related adverse events seen with these therapies. Balancing anti-tumor immunity against autoimmunity remains a major challenge in cancer immunotherapy. Cell-mediated immunity provides defense against intracellular pathogens that cannot be reached by antibodies, viruses replicating inside cells, bacteria like Mycobacterium tuberculosis that survive in phagosomes, protozoan parasites like Plasmodium in liver cells. It also provides surveillance against cancer, eliminating transformed cells before they become established tumors. The diversity of CD4 plus T cell responses allows tailoring of immunity to different pathogen types. The generation of memory T cells provides long-lasting protection, but cell-mediated immunity also causes transplant rejection and contributes to autoimmune diseases, reminding us that powerful defenses can become liabilities when misdirected. While T cells coordinate responses and kill infected cells, much of the actual pathogen elimination is performed by antibodies produced by B cells. These antibodies circulate in blood and tissues, binding to pathogens and toxins and marking them for destruction. The generation of these antibodies, their remarkable diversity and specificity, and their multiple mechanisms of action form the subject of our next chapter. Antibodies are among the most versatile and powerful tools in the immune arsenal. These Y-shaped proteins circulate in blood and other body fluids, binding specifically to foreign molecules and marking them for destruction or directly neutralizing their harmful effects. The term humoral immunity refers to this aspect of adaptive immunity from the archaic medical term humor, meaning body fluid, distinguishing it from cell-mediated immunity. But antibodies do not simply appear. They are produced by B lymphocytes in a process that combines genetic creativity, rigorous selection, and fine-tuning to generate protective responses against an essentially limitless variety of pathogens. B cells, like T cells, originate from hematopoietic stem cells in the bone marrow. Unlike T cells, which leave the marrow and complete development in the thymus, B cells mature entirely in the bone marrow, hence the designation B for bone marrow, though the letter originally stood for bursa of Fabricius, the organ where B cells develop in birds. During development, B cells undergo V-DJ recombination to generate their B cell receptors, which are membrane-bound antibody molecules. Each B cell rearranges its immunoglobulin genes to produce a unique antibody with a specific antigen binding site, and it expresses approximately 100,000 copies of this antibody on its surface. The structure of an antibody is elegant and modular. The basic unit consists of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains, bound together by disulfide bonds and folded into a characteristic Y shape. Each arm of the Y contains one complete antigen-binding site, formed by the variable regions of one heavy chain and one light chain. The stem of the Y, The constant region of the heavy chains determines the antibody's effector functions, whether it can activate complement, bind to Fc receptors on phagocytes, cross the placenta, or be secreted into mucus. The variable regions of antibody chains are generated through VDJ recombination, the same mechanism used for T cell receptors. The heavy chain gene locus contains multiple, variable, diversity, and joining segments. During B cell development, one of each segment type is selected and joined together, creating enormous combinatorial diversity. The light chain loci, either kappa or lambda, have V and J segments but no D segments. Further diversity is generated through imprecise joining, where nucleotides are added or deleted at segment junctions, and through somatic hypermutation, which occurs later during immune responses. The result is a potential repertoire of 10, 11, or more different antibody specificities, vastly exceeding the number of B cells in the body, estimated at 10, 9 to 10, 10. Not all possible antibodies are present at any given time, but the diversity is sufficient to respond to essentially any antigen. This is why vaccination works. Introducing an antigen, even one never encountered by humans, and elicits antibody responses because B cells with receptors capable of binding that antigen almost certainly exists somewhere in the body. Naive B cells, those that have never encountered their specific antigen, circulate through blood and lymphoid tissues, surveying for antigen. Most B cells spend their lives without ever finding their antigen, eventually dying after a few months. But when a B cell's receptor binds to its specific antigen, whether free in solution or displayed on the surface of an antigen-presenting cell like a dendritic cell or macrophage, the B cell becomes activated. For most antigens, full B cell activation requires help from CD4 plus T cells, specifically TFH cells, making these T-dependent antigens. Some antigens, particularly highly repetitive structures like bacterial polysaccharides, can activate B cells without T cell help. These are T-independent antigens. For T-dependent antigens, the activation process is complex and occurs primarily in secondary lymphoid organs. The B cell binds antigen through its surface antibody, internalizes the antigen-antibody complex through receptor-mediated endocytosis, and processes the antigen in endosomes. Peptides derived from the antigen are loaded onto MHC class II molecules and displayed on the B cell surface. Meanwhile, dendritic cells that have captured the same antigen migrate to the lymph node and activate CD4 plus T cells, which differentiate into TFH cells. These TFH cells migrate to the border between the T cell zone and B cell follicles. When a B cell presenting antigen on MHC class II encounters a TFH cell specific for that antigen, the two cells interact. The TFH cell provides help through CD40L binding to CD40 on the B cell and through secretion of cytokines, particularly IL-21. This help, combined with the initial signal from antigen binding to the B cell receptor, fully activates the B cell. It begins proliferating and differentiates into either a plasma cell or a germinal center B cell. Some activated B cells immediately become plasma cells, short-lived antibody-secreting cells that produce IgM, the first antibody class to appear during an immune response. These early plasma cells provide a rapid but relatively low affinity antibody response that helps control infection while the germinal center response develops. Plasma cells are dramatically different from naive B cells. They are larger, with abundant rough endoplasmic reticulum filling the cytoplasm, visible as basophilic staining under the microscope. This extensive protein synthesis machinery allows them to secrete thousands of antibody molecules per second, an astonishing production rate. Other activated B cells, with continued TFH cell help, migrate into the center of B cell follicles and initiate the germinal center reaction, one of the most remarkable processes in immunology. The germinal center is a specialized microenvironment within the B cell follicle where B cells undergo rapid proliferation, somatic hypermutation, class switching, and selection. This process, taking 2 to 3 weeks, generates high affinity antibodies optimally suited to combating the particular pathogen. The germinal center has two distinct zones. The dark zone contains rapidly dividing B cells, called centroblasts, proliferating every 6 to 8 hours. During this proliferation, an enzyme called activation-induced cytidine deaminase, or AIDE, introduces mutations into the variable regions of the immunoglobulin genes at an extraordinarily high rate, about 1 mutation per 1,000 base pairs per cell division, roughly a millionfold higher than the normal mutation rate. This somatic hypermutation randomly alters antibody binding sites, creating variants with different affinities for antigen. Centroblasts then migrate to the light zone and become centrocytes, which stop dividing and test their mutated antibodies. Follicular dendritic cells in the light zone display antigen on their surface, held in antibody-antigen complexes. Centrocytes compete to bind this antigen using their surface antibodies. Those with high-affinity antibodies successfully capture antigen, internalize and process it, and present peptides on MHC class II to TFH cells. These receive survival signals and either return to the dark zone for more proliferation and mutation or differentiate into plasma cells or memory B cells. Centrocytes with low affinity antibodies, or those whose mutations have rendered their antibodies non-functional, cannot capture antigen and present it to TFH cells. Without T cell help, they undergo apoptosis within hours. Their remains are phagocytosed by tingible body macrophages, specialized macrophages in germinal centers that give these structures a characteristic starry sky appearance under the microscope. This ruthless selection ensures that only B cells, with improving or at least maintaining antibody affinity survive, driving affinity maturation. The iterative cycles of proliferation, mutation, and selection in germinal centers can continue for weeks, with antibody affinity increasing 10 to 100-fold or more. This explains why antibody responses improve over time. The first antibodies produced are relatively low affinity, but as germinal centers mature, progressively higher affinity variants are selected. It also explains why booster vaccinations enhance immunity. They drive additional rounds of affinity maturation, producing even better antibodies. During the germinal center reaction, B cells also undergo class switch recombination, changing which constant region their antibodies have while maintaining the same antigen binding variable regions. Naive B cells initially produce IgM and ID, which have identical variable regions, but different constant regions. Through class switching, a B cell can change to produce IgG, IgA, or IgE, altering the antibody's effector functions without changing its specificity. The class switch decision is influenced by cytokines from TFH cells. IFN gamma promotes switching to IgG subclasses, particularly IgG2A in mice, or IgG1 in humans, which are effective at activating complement and binding to Fc receptors on phagocytes. IL-4 promotes switching to IgE, important for anti-parasite immunity, but also responsible for allergies. TGF-beta promotes switching to IgA, the predominant antibody in mucosal secretions. The same enzyme, AID, that causes somatic hypermutation also mediates class switch recombination, introducing breaks in switch regions upstream of each constant region gene segment. The different antibody classes have distinct properties and functions. AGM is a pentamer, 5 antibody units linked together, making it large and unable to leave blood vessels easily. But its multiple binding sites make it effective at agglutinating pathogens, clumping them together, and it is the most efficient antibody at activating complement. IgM is the first antibody produced in primary immune responses and is often used as a marker of recent infection. IgG is the most abundant antibody in blood and the longest lasting, with a half-life of about 21 days compared to 5 days for IgM. IgG can leave blood vessels and enter tissues, providing systemic immunity. It activates complement, though less efficiently than IgM, and binds to Fc receptors on phagocytes, opsonizing pathogens for enhanced uptake. IgG is the only antibody that crosses the placenta, providing passive immunity to fetuses and newborns. There are 4 IgG subclasses in humans, each with slightly different properties. IgA is the predominant antibody in mucosal secretions, saliva, tears, breast milk, and the fluid covering respiratory and gastrointestinal epithelium. Secretory IgA is actually a dimer, 2 antibody units linked with a J chain and associated with a secretory component that protects it from proteolytic degradation. IgA prevents pathogen adhesion to epithelial cells and neutralizes toxins in the gut lumen. It does not activate complement or promote inflammation, making it ideal for mucosal sites where inflammation would be problematic. IgE is present in blood at very low concentrations, but has high affinity for Fc receptors on mast cells and basophils. Even in the absence of antigen, IgE binds to these cells, becoming cell surface bound. When antigen cross-links bound IgE molecules, the cells degranulate, releasing histamine and other mediators. This is the mechanism of immediate hypersensitivity reactions, both the protective responses against parasites and the pathological responses in allergies and anaphylaxis. IgE also binds to eosinophils, promoting antibody-dependent cell-mediated cytotoxicity against large parasites. IgD is expressed on naive B cells alongside IgM, but its function remains somewhat unclear. It may play a role in B cell activation, but mice lacking IgD have largely normal immune function. This is one of several reminders that not everything about the immune system is fully understood despite decades of intensive research. Antibodies combat pathogens through multiple mechanisms, collectively called effector functions. Neutralization is the simplest. Antibodies bind to viruses or bacterial toxins, blocking their ability to attach to and enter cells. An antibody bound to a virus particle prevents the virus from binding to cellular receptors and initiating infection. Antibodies against bacterial toxins like tetanus toxin or diphtheria toxin prevent the toxins from binding to cells and causing damage. Neutralization requires only the antibody's binding function, not interactions with other immune components, and is particularly important in mucosal immunity, where IgA neutralizes pathogens before they cross epithelial barriers. The distinction between primary and secondary antibody responses illustrates the power of immunological memory. Primary responses, occurring upon first encounter with an antigen, take 7 to 10 days to produce substantial antibody levels, initially produce IgM, and generate relatively low affinity antibodies. Secondary responses, upon re exposure to the same antigen, begin within 2 to 3 days, predominantly produce IgG or other class switched antibodies, reach higher peak levels, and produce high affinity antibodies. This faster, stronger, and better response is why second infections with the same pathogen are often asymptomatic or much milder than the first. Antibodies are not just useful in natural immunity, they are powerful therapeutic agents. Passive immunization, the transfer of preformed antibodies, provides immediate but temporary protection. Maternal antibodies transferred across the placenta and in breast milk protect infants. Antibody preparations like immunoglobulin, pooled from thousands of donors, treat immunodeficiency and provide prophylaxis after certain exposures. Hyperimmune globulin, antibodies from individuals who have recovered from specific infections or been vaccinated, treats rabies, tetanus, and other conditions where immediate protection is needed. Monoclonal antibodies, antibodies produced by cloned B cells that are all identical and specific for a single epitope, have revolutionized medicine. Hybridoma technology, developed in the 1970 seconds, fuses antibody-producing B cells with immortal myeloma cells, creating hybrid cells that produce large quantities of specific antibodies indefinitely. More recently, antibodies can be generated through recombinant DNA technology, creating fully human antibodies and modifying them for enhanced properties. Therapeutic monoclonal antibodies treat cancer, autoimmune diseases, infectious diseases, and other conditions. Rituximab targets CD20 on B cells, depleting them to treat B-cell lymphomas and autoimmune diseases. Trastuzumab targets HER2 on breast cancer cells. Adalimumab and other TNF inhibitors treat inflammatory diseases. Antibodies against SARS-CoV-2 were developed as COVID-19 treatments. Checkpoint inhibitors like pembrolizumab, which block PD-1, enhance anti-tumor immunity. The list grows continually as new targets are identified and validated. Antibody engineering has enhanced therapeutic efficacy, humanizing mouse antibodies by replacing most of the mouse sequences with human sequences reduces immunogenicity. Creating bispecific antibodies that bind two different targets simultaneously allows novel functions like bringing T cells into contact with tumor cells. Antibody-drug conjugates link antibodies to toxic chemicals, delivering chemotherapy specifically to cancer cells expressing the target antigen. These innovations demonstrate how understanding antibody structure and function enables therapeutic design. Despite their power, antibodies have limitations. They cannot reach intracellular pathogens, requiring cell-mediated immunity for those. Their large size limits tissue penetration. Production requires B cells, which are absent or defective in certain immunodeficiencies. Evolution of antigen variants, as seen with influenza and HIV, can create escape mutants that existing antibodies don't recognize. And inappropriate antibody responses cause disease. Autoantibodies in autoimmune diseases, IgE in allergies, immune complexes depositing in tissues. Antibodies also mediate certain adverse drug reactions and transfusion reactions. Drug-induced immune thrombocytopenia occurs when antibodies against drug platelet complexes cause platelet destruction. Hemolytic disease of the newborn occurs when maternal antibodies cross the placenta and destroy fetal red blood cells expressing paternal antigens the mother lacks. These remind us that antibodies, like all immune mechanisms, can harm as well as protect. The coordination between T cells and B cells is essential for optimal antibody responses. T cells provide help that drives germinal center reactions, affinity maturation, and class switching. B cells present antigen to T cells, influencing T cell activation and differentiation. Cytokines from T cells determine which antibody classes are produced. Mutual regulation ensures that responses are appropriate to the pathogen type. This intimate cooperation between the cellular and humoral arms of adaptive immunity reflects their coevolution and functional integration. Humoral immunity's ultimate achievement is immunological memory, the ability to respond rapidly and robustly to previously encountered pathogens. Memory B cells and long-lived plasma cells maintain vigilance long after infection has been cleared. This memory is basis of vaccination, one of medicine's greatest successes, and the reason many infectious diseases that once killed millions are now rare or eradicated. It is to immunological memory and its applications that we turn in the next chapter. The immune system's ability to remember previous encounters with pathogens and respond more effectively upon re-exposure is perhaps its most remarkable feature. This immunological memory is not metaphorical, but literal. Specific cells persist for years or decades, maintaining the capacity to recognize particular antigens and mount rapid responses. Memory is what distinguishes adaptive from innate immunity and what makes vaccination possible. Understanding how memory is generated, maintained, and sometimes lost illuminates both the power and limitations of immune protection. The concept of immunological memory has ancient roots. The Greek historian Thucydides, writing about the plague of Athens in 430 BCE, noted that those who recovered from the disease could care for the sick without fear of reinfection. For millennia, this observation remained largely anecdotal. Variolation, the practice of deliberately exposing individuals to material from smallpox lesions to induce mild disease and subsequent immunity, was practiced in China and India for centuries before reaching Europe in the 18th century. But scientific understanding of memory awaited the development of immunology in the late 19th and early 20th centuries. The cellular basis of immunological memory was debated for decades. Early immunologists recognized that antibody responses were faster and stronger upon re-exposure to antigens, but whether this reflected persistent antibodies, long-lived cells, or some other mechanism was unclear. We now know that memory involves multiple components: long-lived memory T and B cells that can respond rapidly to antigen, long-lived plasma cells that continuously secrete antibodies, and perhaps tissue-resident memory cells that guard sites of previous infection. Memory T cells, as discussed in Chapter 10, are heterogeneous. Central memory T cells reside in lymphoid tissues and have high proliferative potential, but require activation and expansion before performing effector functions. Effector memory T cells circulate through peripheral tissues and can immediately produce cytokines or kill targets upon antigen encounter, providing rapid protection but with less proliferative capacity. Tissue-resident memory T cells remain in peripheral tissues like skin, lung, and gut, cannot recirculate, but provide immediate local protection. The distribution and properties of memory T cell subsets determine the kinetics and location of recall responses. Memory B cells are likewise diverse. Some circulate, continuously surveying for their antigen in blood and lymphoid tissues. Others reside in mucosal tissues or spleen. Upon antigen encounter, memory B cells rapidly differentiate into plasma cells, often without needing to re enter germinal centers, producing high affinity antibodies within days. Some memory B cells can also re enter germinal centers upon antigen re exposure, undergoing additional rounds of somatic hypermutation and affinity maturation, further improving antibody quality. Long-lived plasma cells, residing primarily in bone marrow, but also in spleen and mucosal tissues, continuously secrete antibodies without requiring antigen stimulation. These cells provide baseline antibody levels that can neutralize pathogens immediately upon exposure, potentially preventing infection entirely. The longevity of plasma cells varies. Some survive only weeks, while others persist for decades or even lifetimes. What determines plasma cell lifespan and whether it can be therapeutically manipulated are active research questions with implications for vaccine design. The maintenance of immunological memory over long periods raises interesting biological questions. Memory T cells must persist, while naive T cells die after months, requiring ongoing proliferation signals. This is achieved through homeostatic proliferation, slow cell division driven by cytokines like EL7 and EL15 that maintain memory cell numbers without causing full activation. Memory cells also have distinct metabolic programs compared to naive or effector cells, relying more on fatty acid oxidation than glycolysis, which may contribute to their longevity. The durability of memory varies by pathogen and by individual. Measles infection generates lifelong immunity in most individuals, with antibody levels remaining protective for decades. Influenza immunity wanes more quickly, though not entirely, partly due to antigenic drift, where the virus evolves to evade existing antibodies. Tetanus immunity from vaccination requires boosters every 10 years to maintain protective antibody levels, perhaps because Clostridium tetani toxin induces relatively short-lived plasma cells. Understanding why some infections generate durable memory while others do not is crucial for vaccine development. Vaccination exploits immunological memory by exposing the immune system to antigens in a controlled manner, generating memory without causing disease. Edward Jenner's demonstration in 1796 that inoculation with cowpox protected against smallpox is considered the first scientific vaccination, though the underlying principles were not understood for another century. Louis Pasteur later demonstrated that attenuated or weakened pathogens could induce immunity without causing severe disease, and he coined the term vaccination in honor of Jenner's work. Modern vaccines employ several strategies. Live attenuated vaccines use pathogens that have been weakened through laboratory culture, often by growing them in non-human cells or at non-optimal temperatures until they accumulate mutations that reduce virulence. These weakened pathogens can still replicate, triggering robust immune responses similar to natural infection, but without causing serious disease. Live attenuated vaccines typically provide strong, durable immunity, often requiring only 1 or 2 doses. Examples include measles, mumps, rubella, and yellow fever vaccines. The advantage of live attenuated vaccines is their ability to generate strong cellular and humoral immunity, including memory T cells and tissue-resident memory. The replicating pathogen provides sustained antigen exposure, prolonged inflammation, and activation of innate immunity, all of which enhance memory generation. However, live vaccines carry risks. They can cause disease in immunocompromised individuals, they may revert to virulence, and they require careful storage to maintain viability. These limitations have driven development of alternative approaches. Inactivated vaccines use pathogens that have been killed by heat, chemicals, or radiation, rendering them unable to replicate but preserving their antigens. Polio, hepatitis A, and influenza vaccines are available in inactivated forms. These vaccines are safer than live vaccines, particularly for immunocompromised individuals, but typically generate weaker immune responses, especially T cell responses, requiring multiple doses and booster vaccinations to maintain immunity. Adjuvants, substances that enhance immune responses, are often added to improve efficacy. Subunit vaccines contain only specific pathogen components, typically surface proteins, rather than the whole organism. Hepatitis B vaccine contains only the viral surface antigen, produced in yeast through recombinant DNA technology. Human papillomavirus, or HPV, vaccines contain virus-like particles, protein shells that resemble the virus structurally but lack genetic material and cannot replicate. Subunit vaccines are safe and well tolerated, but require adjuvants to generate adequate immunity. They typically induce strong antibody responses, but weaker T cell responses compared to live vaccines. Conjugate vaccines address a particular challenge: the immune response to polysaccharides. Young children respond poorly to polysaccharide antigens because these T-independent antigens don't efficiently activate naive B cells or generate memory in children under 2 years old. Conjugate vaccines link polysaccharides to protein carriers, converting them into T-dependent antigens that elicit robust, lasting immunity even in infants. Haemophilus influenzae type B, or Hib, pneumococcal, and meningococcal conjugate vaccines have dramatically reduced bacterial meningitis in children. mRNA vaccines represent a newer approach, though the concept has been studied for decades. These vaccines contain messenger RNA encoding pathogen antigens, typically encapsulated in lipid nanoparticles for delivery into cells. Once inside cells, the mRNA is translated into protein, which is then processed and presented on MHC molecules, activating both B and T cell responses. The COVID-19 pandemic saw the first large-scale deployment of mRNA vaccines, with Pfizer-BioNTech and Moderna vaccines proving highly effective and safe. mRNA vaccines offer several advantages: rapid development and manufacturing once antigen sequences are known, no risk of infection since they contain no live pathogen, and ability to induce both antibody and T cell responses. They also have limitations. The mRNA is unstable, requiring ultra-cold storage for some formulations, and the lipid nanoparticles can cause inflammatory side effects. Whether mRNA vaccines will become standard for other diseases remains to be seen, but the technology has been validated. Adjuvants enhance vaccine responses by activating innate immunity and creating a favorable environment for adaptive immune responses. Aluminum salts, or alum, have been used for nearly a century despite incompletely understood mechanisms. They create depots that slowly release antigen, activate complement, and promote uptake by antigen-presenting cells. Newer adjuvants include oil and water emulsions, toll-like receptor agonists like monophosphoryl lipid, derived from bacterial LPS, and other innate immune stimulants. The choice of adjuvant influences the type of immune response generated, allowing vaccines to be tailored for specific needs. Vaccine schedules optimize immunity through strategic timing of doses. Most vaccines require multiple doses to generate adequate immunity, with intervals between doses allowing immune responses to mature. Booster doses years after initial vaccination reinforce memory and maintain protective antibody levels. The timing is not arbitrary, but based on immunological principles, allowing time for germinal center reactions to complete and memory to form, but boosting before antibody levels fall below protective thresholds. Herd immunity, or community immunity, represents an emergent property of vaccination at the population level. When enough individuals are immune, pathogen transmission is interrupted even if some individuals remain susceptible. The percentage of the population that must be immune to achieve herd immunity varies by disease, depending on how contagious the pathogen is. Measles, extremely contagious, requires 95% immunity. Less contagious pathogens may require 70 to 80%. Herd immunity protects those who cannot be vaccinated, infants too young for vaccination, individuals with contraindications, and immunocompromised people who don't respond adequately to vaccines. The success of vaccination is difficult to overstate. Smallpox has been eradicated, the only human disease ever eliminated, through coordinated global vaccination campaigns. Polio has been eliminated from most of the world and is close to eradication. Measles, mumps, rubella, and other childhood diseases that once killed or disabled millions are now rare in countries with high vaccination coverage. Vaccines prevent an estimated 4 to 5 million deaths annually worldwide. Yet vaccination faces challenges. Vaccine hesitancy, fueled by misinformation and distrust of institutions, threatens herd immunity in some communities, leading to outbreaks of preventable diseases. The 1,998 publication of a fraudulent paper falsely linking MMR vaccine to autism, though thoroughly debunked and retracted, caused lasting damage to vaccine confidence. Addressing vaccine hesitancy requires not just scientific communication, but understanding of social, cultural, and psychological factors that influence vaccine acceptance. Some pathogens evade vaccine-induced immunity through antigenic variation, changing their surface proteins so existing antibodies no longer recognize them. Influenza undergoes both antigenic drift, gradual accumulation of mutations, and antigenic shift, reassortment of gene segments from different strains, requiring annual updating of flu vaccines. HIV's extraordinary mutation rate and ability to integrate into host genomes has so far prevented effective vaccine development despite decades of effort. Malaria's complex life cycle and ability to hide from antibodies within infected cells pose similar challenges. Therapeutic vaccination, using vaccines to treat rather than prevent disease, is being explored for chronic infections and cancer. Therapeutic HPV vaccines aim to eliminate existing HPV infections and associated precancerous lesions. Cancer vaccines attempt to generate or enhance immune responses against tumors. Some use tumor antigens as immunogens. Others use dendritic cells loaded with tumor material. Checkpoint inhibitors can be considered a form of therapeutic vaccination, releasing brakes on existing anti-tumor immunity. While most cancers remain resistant to vaccination, advances in understanding tumor immunology may yet make therapeutic cancer vaccines feasible. The COVID-19 pandemic demonstrated both the power and limitations of modern vaccine science. Multiple effective vaccines were developed in record time through unprecedented scientific collaboration, regulatory flexibility, and financial investment. Vaccines prevented millions of deaths and reduced disease severity even as viral variants emerged. Yet vaccine distribution was grossly inequitable, with wealthy nations accumulating doses while poor nations waited, ethical and practical failures that prolonged the pandemic. The pandemic also intensified vaccine hesitancy in some populations while demonstrating that rapid vaccine development is possible when resources are dedicated. Memory is not permanent, at least not always. Antibody levels decline over time, memory cells may not persist indefinitely, and pathogens evolve to evade immunity. But for many infections, memory persists long enough to prevent reinfection or to ensure that reinfections are mild. This is the goal of vaccination: to generate memory that provides lasting protection, or at least protection long enough that boosters can maintain it. Understanding what factors determine memory duration and quality is critical for designing better vaccines. Immunological memory is not unique to adaptive immunity. Trained immunity, or innate immune memory, has been recognized in recent years. Innate immune cells exposed to certain stimuli show enhanced responsiveness to subsequent challenges, an effect mediated by epigenetic modifications rather than genetic changes. BCG vaccination provides protection beyond just tuberculosis, possibly through trained immunity. This challenges traditional distinctions between innate and adaptive immunity and may offer new approaches to enhancing vaccine responses. The immune system's ability to remember is both its greatest strength and a source of potential problems. Memory to pathogens protects against reinfection. Memory to self-antigens, when it occurs, drives autoimmune disease. Memory to allergens causes allergies. Memory to transplanted organs causes rejection. Memory to tumor antigens, when it can be generated, provides cancer immunosurveillance. Modulating memory, enhancing it where desired, and suppressing it where harmful is a central goal of immunology research. Vaccines work by harnessing the immune system's natural ability to remember, but pathogens also have strategies to evade immunity, including memory. These evolutionary conflicts between immune defenses and pathogen countermeasures shape both host and pathogen biology, driving their coevolution over millions of years. It is to these immune evasion strategies, the ways that pathogens fight back against immunity, that we now turn our attention. The immune system is formidable, but pathogens are not passive victims. They have been evolving for billions of years, developing strategies to evade, subvert, or exploit immune defenses. This evolutionary arms race between host immunity and pathogen evasion has shaped both organisms profoundly. Every immune mechanism has spawned countermechanisms, and every pathogen success has driven immune adaptation. Understanding immune evasion illuminates both the sophistication of pathogen biology and the vulnerabilities in immune defenses that might be therapeutically exploited. The simplest evasion strategy is avoiding recognition entirely. Many bacteria possess capsules, thick layers of polysaccharides surrounding the cell wall that interfere with complement deposition and phagocytosis. Streptococcus pneumoniae and Haemophilus influenzae cause severe infections in part because their capsules inhibit complement-mediated opsonization and resist phagocytosis. The capsule physically separates the bacterial surface from antibodies and complement proteins, and its anionic charges may electrostatically repel negatively charged immune molecules. Notably, conjugate vaccines that generate antibodies specifically against capsular polysaccharides can overcome this evasion. Mycobacterium tuberculosis has evolved a complex waxy cell wall rich in mycolic acids that resists degradation by lysosomal enzymes. When phagocytosed by macrophages, these bacteria survive in phagosomes by preventing phagosome lysosome fusion, creating an intracellular niche sheltered from most immune effectors. The bacteria can persist in macrophages for years or decades, causing latent tuberculosis infection that can reactivate if immunity wanes. Only a small fraction of infected individuals develop active disease, but globally, latent TB represents an enormous reservoir of infection. Listeria monocytogenes employs a different strategy. It escapes the phagosome entirely. This bacterium produces a toxin, Listeria lysin O, that forms pores in the phagosome membrane, allowing the bacteria to escape into the cytoplasm. There, sheltered from lysosomal killing mechanisms, Listeria replicates. The bacteria even hijack the host cell's actin cytoskeleton, forming actin tails that propel them into adjacent cells, allowing cell-to-cell spread without exposure to extracellular immunity. This intracellular lifestyle makes Listeria infections particularly difficult to clear and explains why cell-mediated immunity, especially Fn gamma, activated macrophages, and CD8 plus T cells, is essential for control. Viruses face a particular challenge. They must replicate inside cells, where MHC class I presentation should expose them to CD8 plus T cells. Many viruses have evolved mechanisms to interfere with MHC class I presentation. Human cytomegalovirus, or HCMV, encodes multiple proteins that disrupt antigen presentation. US3 retains MHC class I molecules in the endoplasmic reticulum, preventing surface expression. US2 and US11 export MHC class I molecules from the ER into the cytosol, where they are degraded by proteasomes. US6 blocks the TAP transporter, preventing peptide entry into the ER. This multi-pronged attack on MHC class I expression allows HCMV infected cells to evade CD8 plus T cell recognition. Herpes simplex virus uses a similar but distinct approach. Its ICP47 protein binds to TAP and blocks peptide transport. Adenoviruses encode E3 over 19K, which retains MHC class I in the ER. HIV's NEF protein downregulates MHC class I surface expression. These diverse mechanisms converge on the same goal, reducing MHC class I levels to avoid T cell detection. However, as discussed previously, this creates vulnerability to NK cells, forcing viruses to balance T cell evasion against NK cell susceptibility. Some viruses encode MHC class I mimics or NK cell inhibitory ligands to address this dilemma. Interfering with interferons represents another common viral strategy. Influenza virus NS, one protein inhibits interferon production and blocks interferon signaling. Hepatitis C virus protease cleaves MAVs, an adapter protein essential for interferon induction. Pox viruses encode soluble interferon receptors that bind and neutralize interferons before they can signal to cells. These mechanisms allow viruses to replicate before strong antiviral states can be established, buying time to complete their life cycles and spread to new hosts. Antigenic variation, changing surface antigens to evade existing immunity, is employed by numerous pathogens. Influenza virus undergoes antigenic drift through accumulation of point mutations in hemagglutinin and neuraminidase, the major surface glycoproteins recognized by antibodies. These mutations occur continuously during viral replication, and variants that escape existing antibodies have a selective advantage. This necessitates annual reformulation of influenza vaccines to match circulating strains, a perpetual game of catch up between vaccine design and viral evolution. Influenza can also undergo antigenic shift through reassortment of gene segments from different viral strains, potentially creating pandemic strains with novel surface antigens to which population immunity is absent. The 1918, 1957, 1968, and 2009 pandemics all involved antigenic shift. While drift is gradual, shift can be abrupt, radically changing the virus's antigenic properties and evading all existing immunity. This capacity for radical antigenic change makes influenza a persistent pandemic threat despite decades of vaccination. HIV takes antigenic variation to an extreme. Its reverse transcriptase lacks proofreading, introducing mutations at an extraordinary rate, approximately one mutation per genomic replication. With multiple replication cycles daily in an infected individual, HIV generates extensive genetic diversity, creating quasi-species, populations of related but distinct viral variants. Antibodies that neutralize one variant are evaded by others carrying mutations in antibody-binding epitopes. This antigenic diversification occurs even within a single infected individual, frustrating immune responses and vaccine development. African trypanosomes, parasites causing sleeping sickness, employ a particularly elegant variation strategy called antigenic variation. These unicellular parasites coat their surface with variant surface glycoproteins, or VSGs. At any time, a trypanosome expresses only one of hundreds of VSG genes. When the host mounts antibody responses against the dominant VSG, rare trypanosomes that have switched to expressing a different VSG escape antibody recognition and proliferate. This creates waves of parasitemia as the immune system successively controls each VSG variant, only to be evaded by the next. Chronic infection can persist for years as the parasite cycles through its VSG repertoire. Plasmodium falciparum, the malaria parasite, employs multiple evasion strategies across its complex life cycle. In its liver stage, the parasite resides in hepatocytes, largely hidden from immune surveillance. In the blood stage, infected erythrocytes express variant proteins on their surface that allow them to adhere to vascular endothelium, sequestering them away from splenic filtration. The parasite also undergoes antigenic variation through differential expression of VAR genes encoding pFEMP1 proteins, evading antibody responses. Chronic malaria involves ongoing cycles of parasite multiplication and immune control without complete clearance. Molecular mimicry involves pathogens expressing molecules that resemble host molecules, exploiting immune tolerance to self. Streptococcus pyogenes, group A Streptococcus, produces M protein that resembles cardiac myosin. In some individuals, antibodies against M protein cross-react with cardiac tissue, causing rheumatic fever and rheumatic heart disease. This demonstrates how pathogen mimicry can trigger autoimmunity, with the immune system attacking host tissues while attempting to eliminate pathogens. Some pathogens actively suppress immune responses. Measles virus infects lymphocytes and dendritic cells, causing lymphopenia and immunosuppression that can last weeks to months after acute infection. This immune suppression increases susceptibility to secondary infections, historically a major cause of measles mortality. HIV's destruction of CD4 plus T cells progressively cripples adaptive immunity, creating the profound immunodeficiency of AIDS. Mycobacterium leprae infects Schwann cells in peripheral nerves and suppresses local immune responses, allowing chronic infection that can persist for years. Helminthic worms, too large to be phagocytosed or killed by conventional mechanisms, employ multiple evasion strategies. They secrete molecules that suppress Th1 responses and promote Th2 responses, which, while generating eosinophils and IgE, are less effective at worm expulsion than Th1 responses would be. Some worms coat themselves with host proteins, molecular camouflage that makes them resemble self. Others shed their surface proteins, decoys that absorb antibodies and complement away from the intact worm. And their long lifespans, sometimes decades within the host, demonstrate the success of these strategies. Biofilms represent a collective evasion strategy. Many bacteria can form biofilms, communities of bacteria embedded in a self-produced extracellular matrix of polysaccharides, proteins, and DNA. Within biofilms, bacteria are protected from antibodies, complement, and phagocytes, which cannot penetrate the matrix effectively. Bacteria in biofilms also exhibit altered metabolism and can enter dormant states resistant to antibiotics. Biofilms form on medical devices like catheters and implants, causing persistent infections difficult to eradicate without device removal. Chronic biofilm infections, like those in cystic fibrosis lungs, resist immune clearance despite ongoing inflammation that damages tissues. Latency, entering a dormant state where the pathogen remains quiescent and largely invisible to the immune system, is employed by several viruses. Herpes viruses establish latent infections in neurons or lymphocytes, maintaining viral genomes with minimal gene expression. Periodic reactivation allows viral replication and spread to new hosts. Varicella zoster virus, causing chickenpox in primary infection, establishes latency in sensory ganglia and can reactivate decades later as shingles. Epstein-Barr virus establishes lifelong latent infection in B cells, periodically reactivating to produce infectious particles in saliva. During latency, viral antigen expression is minimal, providing little for the immune system to target. Memory T cells specific for lytic viral antigens don't recognize latently infected cells. Only when reactivation occurs, restoring viral replication and antigen production, can immune responses control the infection. This balance between latency and reactivation, influenced by immune status, stress, and other factors, allows persistent infections that can transmit intermittently over the host's lifetime. Immune subversion goes beyond simple evasion. Some pathogens actively manipulate host immunity to their advantage. Cytomegalovirus encodes a viral IL-10 homologue that suppresses inflammatory responses. Poxviruses produce chemokine-binding proteins that neutralize host chemokines, reducing leukocyte recruitment. Epstein-Barr virus encodes a viral IL-10 and other immunomodulatory molecules. By producing molecules that resemble or interact with host immune regulators, these pathogens co-opt immune machinery for their purposes. Toxoplasma gondii, a protozoan parasite, actually modifies host behavior, potentially to favor transmission. Infected rodents lose their innate fear of cat odors and may be attracted to them, increasing predation risk. Since cats are T. gondia's definitive host, where sexual reproduction occurs, this behavioral modification benefits the parasite. The mechanisms likely involve parasite cysts in brain regions controlling fear and alterations in neurotransmitter systems. Whether similar behavioral effects occur in infected humans remains controversial but concerning, given that roughly one-third of the human population carries latent toxoplasma infection. The study of immune evasion has also revealed fundamental principles of immunity. The discovery that some viruses inhibit MHC class I led to understanding NK cells' role in recognizing missing self. Studies of antigenic variation clarified how antibody diversity is generated and selected. Investigation of helminth immunomodulation revealed each two responses and IgE function. Pathogen evasion mechanisms have thus served as tools for dissecting immune function. Some immune evasion strategies verge on the remarkable. Herpes simplex virus glycoprotein C binds complement component C3b and acts as a cofactor for factor I, which inactivates C3b, essentially hijacking the host's complement regulation system. Staphylococcus aureus produces protein A, which binds the Fc region of antibodies in the wrong orientation, preventing complement activation and opsonization while making the bacteria appear antibody-coated. Yersinia pestis, plague bacterium, injects effector proteins through a type 3 secretion system directly into host cells, where they disrupt cytoskeletal dynamics and prevent phagocytosis. These mechanisms are not just fascinating biology, they are also potential therapeutic targets. If we can block immune evasion, we might restore immune control without needing to directly kill pathogens. This concept underlies some antimicrobial strategies, targeting bacterial capsule synthesis, inhibiting viral proteins that block interferon signaling, or using adjuvants that overcome immune suppression. The principle is to let the immune system do what it evolved to do by removing the obstacles pathogens have evolved. The evolutionary perspective also cautions against assuming immune evasion is always successful or permanent. Most infections are eventually cleared, indicating that immunity usually prevails despite evasion mechanisms. Pathogens that evade too effectively may kill their hosts before transmitting, selected against. Pathogens that trigger too little immunity may be cleared before establishing infection. Successful pathogens often achieve a balance, evading enough to persist, but not so much as to prevent host survival and transmission. Chronic infections represent one outcome of this balance. The pathogen persists, often at low levels, with immunity controlling but not eliminating infection. Hepatitis B and C, HIV without treatment, Helicobacter pylori, herpes viruses all establish chronic infections where host and pathogen coexist, sometimes for decades. The host survives, the pathogen transmits, and natural selection favors this equilibrium over acute pathogenic strategies that kill hosts rapidly. Commensalism and mutualism represent endpoints of this evolutionary process. Some organisms that were once pathogens may have evolved to become harmless or beneficial, their immune evasion mechanisms becoming mechanisms of tolerance. The mitochondria in our cells are thought to derive from ancient bacteria that established endosymbiotic relationships with eukaryotic ancestors. The distinction between pathogen and symbiont is not always clear, and immune tolerance to beneficial microbes may have evolved from ancient evasion counter evasion dynamics. Understanding immune evasion thus provides not just a catalog of microbial tricks, but insights into coevolution, the selective pressures shaping life, and the dynamic equilibria between hosts and pathogens. It reminds us that immunity is not a fixed fortress, but an active, evolving system engaged in ongoing conflict with equally evolving adversaries. And it highlights that the immune system, for all its sophistication, is not infallible. Sometimes immunity itself becomes the problem, attacking self instead of pathogens, overreacting to harmless antigens, or failing entirely to function. These immune system failures, our topic in the next chapter, cause substantial human suffering and are the focus of much immunological research and therapeutic development. The immune system is a powerful defense mechanism, but power comes with risk. Systems designed to recognize and destroy threats can malfunction, turning against the body they should protect or overreacting to harmless stimuli. Immune system disorders fall into three broad categories: autoimmunity, where immunity attacks self; hypersensitivity or allergy, where immunity overresponds to harmless antigens; and immunodeficiency, where immunity is inadequate or absent. Together, these disorders affect hundreds of millions of people worldwide and range from mild inconveniences to fatal diseases. Understanding them illuminates both the delicate balance immunity requires and the consequences when that balance fails. Autoimmunity represents a breakdown of self-tolerance, the normally robust mechanisms that prevent immune responses against the body's own tissues. Despite central tolerance in the thymus and bone marrow, where autoreactive lymphocytes are deleted, and peripheral tolerance mechanisms that suppress autoreactive cells escaping central tolerance, autoimmunity occurs in approximately 5 to 10% of the population in developed countries. Why tolerance fails in these individuals remains incompletely understood, but involves genetic susceptibility, environmental triggers, and stochastic events. Type 1 diabetes, an autoimmune disease, involves T cell and antibody mediated destruction of insulin producing beta cells in the pancreatic islets. The disease typically manifests in childhood or adolescence, though it can occur at any age. Autoreactive CD8 plus T cells infiltrate the islets and kill beta cells, aided by inflammatory cytokines from CD4 plus T cells and possibly by antibodies against insulin or other beta cell proteins. The progressive loss of beta cells causes hyperglycemia when insulin production falls below the threshold needed to regulate blood glucose. The cause of type 1 diabetes involves genetic susceptibility, particularly certain HLA types like HLA-DR3 and HLA-DR4, which confer markedly increased risk. These HLA molecules may present beta cell antigens in ways that activate autoreactive T cells. Environmental triggers, possibly viral infections or dietary factors in early childhood, may initiate the autoimmune process in genetically susceptible individuals. Once begun, the autoimmune attack can persist for years before clinical diabetes appears, providing a window for potential intervention, though effective preventive therapies remain elusive. Rheumatoid arthritis involves autoimmune attack on joint synovium, the membrane lining joint cavities. Autoreactive T cells and B cells infiltrate synovial tissue, producing inflammatory cytokines and autoantibodies. Rheumatoid factor, antibodies against the Fc region of IgG, and anti-citrulline protein antibodies, or ACPA, are characteristic findings. These autoantibodies form immune complexes that activate complement and drive chronic inflammation. The inflamed synovium, called pannus, invades cartilage and bone, causing progressive joint destruction, deformity, and disability. Treatment of rheumatoid arthritis has been revolutionized by biologic therapies targeting specific immune components. TNF inhibitors like etanercept, infliximab, and adalimumab block this key inflammatory cytokine, dramatically reducing inflammation and slowing joint destruction. IL-6 inhibitors, rituximab, which depletes B cells, and abatacept, which blocks T cell co-stimulation, also prove effective. These therapies demonstrate that understanding immune mechanisms translates into treatments, though they require careful monitoring as they increase infection risk. Multiple sclerosis is an autoimmune disease of the central nervous system where T cells attack myelin, the insulating sheath around nerve axons. The disease causes relapsing remitting neurological symptoms as inflammation damages myelin in scattered CNS locations, creating the sclerotic plaques visible on MRI. Symptoms depend on lesion location: optic neuritis causing vision loss, spinal cord lesions causing weakness or sensory changes, brainstem lesions causing double vision or coordination problems. Over years, many patients transition to progressive disease with accumulating neurological disability. The cause of multiple sclerosis likely involves molecular mimicry, where immune responses to infectious agents cross-react with myelin proteins. Epstein-Barr virus infection is strongly associated with MS risk, and EBV antigens may resemble myelin antigens. Genetic susceptibility, particularly certain HLA-DRB1 alleles, vitamin D deficiency, and other environmental factors also contribute. Treatments include interferon beta, which has immunomodulatory effects, glatiramer acetate, which may induce regulatory T cells, natalizumab, which blocks lymphocyte entry into the CNS, and other immunosuppressants that reduce disease activity. Systemic lupus erythematosus, or SLE, is a systemic autoimmune disease where antibodies against nuclear components, particularly DNA and ribonucleoproteins, form immune complexes that deposit in multiple organs. These immune complexes activate complement and trigger inflammation in skin, joints, kidneys, blood vessels, and other tissues. The classic malar rash, a butterfly-shaped facial rash, photosensitivity, arthritis, and kidney disease are common manifestations. Severe cases involve life-threatening kidney failure or central nervous system involvement. SLE predominantly affects women of childbearing age, suggesting hormonal influences. Genetic factors, particularly complement deficiencies and certain HLA types, increase risk. Some cases are triggered by medications, resolving when the drug is stopped. Treatment involves corticosteroids to suppress inflammation, hydroxychloroquine, which modulates immune activation, and immunosuppressants for severe disease. Belimumab, an antibody that blocks B-cell survival factor, BAFF, represents a more targeted therapy, though SLE remains challenging to treat. The mechanisms underlying autoimmunity are diverse. Molecular mimicry, where pathogen antigens resemble self-antigens, can trigger cross-reactive immunity. Epitope spreading occurs when initial immune responses damage tissue, releasing previously sequestered antigens that trigger additional autoimmune responses. Bystander activation involves inflammatory cytokines from infections activating autoreactive lymphocytes. Defects in regulatory T cells can impair suppression of autoreactive cells. Loss of immune privilege can expose sequestered antigens to immunity, as in sympathetic ophthalmia, where eye injury triggers autoimmunity against the fellow eye. Genetic susceptibility is evident in autoimmune diseases' familial clustering and associations with specific HLA types and other immune related genes. HLA associations suggest that how self antigens are presented influences autoimmunity risk. Polymorphisms in genes regulating immune activation, like PTPN22 and CTLA4, affect autoimmunity susceptibility. But genetics explains only part of risk. Environmental factors clearly play roles, as evidenced by incomplete concordance in identical twins and rising autoimmune disease incidence in recent decades. Shifting to hypersensitivity disorders, these involve exaggerated immune responses to harmless environmental antigens called allergens. Type 1 hypersensitivity, immediate allergic reactions, involves IgE antibodies bound to mast cells and basophils. When allergen cross-links these cell-bound IgE molecules, the cells degranulate, releasing histamine, leukotrienes, and other mediators, causing rapid onset symptoms, itching, hives, bronchoconstriction, vasodilation, increased vascular permeability. Hay fever, or allergic rhinitis, results from inhaled allergens like pollen or dust mite proteins, triggering IgE-mediated responses in nasal mucosa. Symptoms include sneezing, nasal congestion, itchy eyes, and nose. Food allergies involve IgE against food proteins, causing symptoms ranging from mild oral itching to severe anaphylaxis. Asthma, while multifactorial, often has an allergic component where inhaled allergens trigger bronchoconstriction, mucus hypersecretion, and airway inflammation. Anaphylaxis represents the most severe type 1 hypersensitivity, a systemic reaction that can be fatal. Widespread mast cell degranulation causes vasodilation and increased vascular permeability throughout the body, leading to circulatory shock. Airway edema and bronchoconstriction cause respiratory distress. Common triggers include foods, particularly peanuts, tree nuts, shellfish, insect stings, and medications like penicillin. Treatment requires immediate epinephrine injection to counteract vasodilation and bronchoconstriction, followed by supportive care. The hygiene hypothesis attempts to explain rising allergy prevalence in developed countries. It proposes that reduced microbial exposure in early childhood due to improved hygiene, antibiotics, and lifestyle changes impairs immune system maturation. Without adequate Th1 polarizing infections, the immune system defaults to two responses, promoting IgE production and allergic sensitization. While likely oversimplified, this hypothesis has generated research into how early life exposures shape immune development and allergy risk. Type 2 hypersensitivity involves antibodies binding to cell surface or matrix antigens, causing cell destruction or dysfunction. Hemolytic transfusion reactions occur when antibodies attack transfused red blood cells bearing incompatible blood group antigens. Hemolytic disease of the newborn results from maternal antibodies crossing the placenta and destroying fetal red blood cells, particularly in Rh incompatibility. Goodpasture syndrome involves antibodies against basement membrane collagen, causing glomerulonephritis and pulmonary hemorrhage. Type 3 hypersensitivity involves immune complex deposition, When antibody antigen complexes are not efficiently cleared, they deposit in tissues, activating complement and recruiting neutrophils that cause inflammation and tissue damage. Serum sickness, historically caused by therapeutic animal antisera, results from immune complexes depositing in blood vessels, causing fever, rash, arthritis, and glomerulonephritis. Systemic lupus erythematosus, discussed earlier, is partially a type 3 hypersensitivity disease, given its prominent immune complex pathology. Severe combined immunodeficiency, or SCID, represents the most profound primary immunodeficiency. Affected infants lack functional T and B cells due to defects in lymphocyte development. Without treatment, they suffer severe, recurrent infections and usually die within the first year of life. The bubble boy, David Vetter, who lived in a sterile environment for 12 years, had SCID. Treatment requires hematopoietic stem cell transplantation to reconstitute immunity, and increasingly, gene therapy to correct the underlying genetic defect shows promise. X-linked agamoglobulinemia results from mutations in the BTK gene required for B-cell development. Affected boys, as it's X-linked, lack mature B cells and cannot produce antibodies. They suffer recurrent bacterial infections, particularly of respiratory and gastrointestinal tracts, beginning after maternal antibodies wane around 6 months of age. Treatment involves regular intravenous immunoglobulin replacement, providing antibodies the patients cannot make themselves. Chronic granulomatous disease results from defects in NADPH oxidase, the enzyme complex generating reactive oxygen species in phagocytes. Without oxidative burst, neutrophils and macrophages cannot kill catalase-positive organisms. Patients suffer recurrent bacterial and fungal infections, particularly with Staphylococcus aureus, Aspergillus, and other catalase-positive organisms. Chronic inflammation and granuloma formation cause additional complications. Treatment includes prophylactic antibiotics, antifungals, and interferon gamma. Common variable immunodeficiency, or CVID, is a heterogeneous group of disorders causing hypogammaglobulinemia, low antibody levels. Unlike X-linked agamoglobulinemia, B cells are present but don't differentiate properly into antibody-secreting plasma cells. CVID typically manifests in adolescence or adulthood with recurrent sinus pulmonary infections. Treatment involves immunoglobulin replacement. Some patients also develop autoimmunity or lymphoproliferative disorders, complications that remain poorly understood. HIV, AIDS represents the most significant acquired immunodeficiency. HIV infects CD4 plus T cells, macrophages, and dendritic cells through CD4 and co-receptors CCR5 or CXCR4. The virus integrates into host DNA and can establish latent infection. Over years without treatment, progressive CD4 plus T cell depletion causes immunodeficiency. When CD4 counts fall below 200 cells per microliter, opportunistic infections and cancers define AIDS. Antiretroviral therapy, combining multiple drugs targeting different steps in HIV replication, can suppress viral replication to undetectable levels, allowing immune recovery and near normal life expectancy. However, treatment must be lifelong as latent reservoirs persist. HIV remains incurable with current therapies, though rare cases of cure following stem cell transplantation from donors with CCR5 mutations provide hope. HIV prevention through pre-exposure prophylaxis and treatment as prevention, where suppressing viral loads prevents transmission, are increasingly important control strategies. Immunosenescence, age-related immune decline, affects everyone who lives long enough. Thymic involution reduces new T cell production. The T cell repertoire becomes restricted, dominated by memory cells with limited ability to respond to novel antigens. B cell function declines with reduced antibody responses and quality. Innate immunity shows increased inflammatory activity, chronic low-grade inflammation or inflammaging, while simultaneously showing reduced responses to acute infections. These age-related changes increase susceptibility to infections, reduce vaccine efficacy, and contribute to chronic diseases. Influenza and pneumococcal pneumonia cause substantial mortality in elderly populations. Shingles from varicella zoster reactivation increases with age as cellular immunity wanes. Cancer incidence rises, partly reflecting declining immune surveillance. Understanding and mitigating immunosenescence is increasingly important as populations age. Immune dysfunction, whether autoimmunity, hypersensitivity, or immunodeficiency, demonstrates that immunity requires precise calibration. Too much causes autoimmunity or allergy. Too little causes immunodeficiency. The balance must be maintained across a lifetime despite changing threats, aging, and environmental influences. That this balance usually succeeds is testament to immunity's robust design. That it sometimes fails reminds us of the system's complexity and the ongoing need for research to understand and treat immune disorders. The immune system that we've explored across these chapters exists not in isolation, but as part of a whole organism, influenced by genetics, environment, nutrition, behavior, and age. How we can support immune function throughout life, maintain the delicate balance that protects without harming, and what factors influence immunity, for better or worse. The immune system does not exist in a vacuum. It is influenced by every aspect of an organism's existence: the genes inherited from parents, the nutrients consumed, the pathogens encountered, the sleep obtained, the stress experienced, and the inexorable process of aging. Understanding these influences allows us to support immune function to maintain the guardian that protects us from birth until death. While we cannot fundamentally redesign our immune systems, we can optimize their function through choices and interventions grounded in immunological understanding. Life begins with borrowed immunity. The developing fetus in utero receives some immune protection from the mother, though the placenta's semi-permeability creates a delicate balance. Maternal IgG antibodies cross the placenta, particularly during the third trimester, providing passive immunity to the newborn. These antibodies recognize pathogens the mother has encountered through infection or vaccination, offering protection during the infant's first months when their own immune system is still developing. This is why maternal vaccination, particularly against influenza and pertussis during pregnancy, protects not just the mother, but also the infant. After birth, breastfeeding provides additional immune support. Colostrum, the first milk produced, is rich in IgA antibodies, immune cells, antimicrobial proteins, and growth factors. Secretory IgA coats the infant's gastrointestinal tract, preventing pathogen adhesion and entry. While these maternal antibodies provide only passive, temporary immunity, they protect during a vulnerable period. Breastfed infants have lower rates of gastrointestinal and respiratory infections, and possibly lower rates of allergies and autoimmune diseases, though confounding factors make the latter associations less certain. The infant's own immune system begins developing during fetal life and continues maturing postnatally. At birth, innate immunity is relatively functional, though neonatal neutrophils and macrophages show reduced functional capacity compared to adult cells. Adaptive immunity is naive, lacking the memory that comes from antigen exposure. The thymus is at its peak relative size and activity, generating vast numbers of T cells. B cells are present but naive. Over the first years of life, repeated exposures to antigens through infections, vaccination, and environmental exposures build immunological memory. Early childhood represents a critical period for immune education. Exposure to diverse microbes, both pathogenic and commensal, trains the immune system to distinguish threats from harmless antigens and to calibrate response magnitudes appropriately. The hygiene hypothesis discussed earlier proposes that excessive cleanliness and reduced microbial exposure may impair this education, contributing to allergies and autoimmune diseases. While the evidence remains debated, it seems clear that some microbial exposure is necessary for proper immune development. The microbiome, the community of microorganisms inhabiting our bodies, plays a crucial role in immune education and function. Colonization begins at birth, with delivery mode influencing initial microbial composition. Vaginal delivery exposes infants to maternal vaginal and fecal microbiota, while cesarean delivery provides different initial exposure. Breastfeeding further shapes the microbiome through oligosaccharides that promote beneficial bacteria like bifidobacterium. By age 2 to 3, the gut microbiome stabilizes into an adult-like composition, though it remains dynamic throughout life. The gut microbiome influences immunity profoundly. Commensal bacteria stimulate development of gut-associated lymphoid tissue, promote IgA production, maintain epithelial barrier function, and influence systemic immunity. Segmented filamentous bacteria in mice, and likely similar organisms in humans, induce Th17 cell development. Other bacteria promote regulatory T cell differentiation. Short-chain fatty acids produced by bacterial fermentation of dietary fiber have anti-inflammatory properties and influence T cell differentiation. Disrupting the microbiome with antibiotics can alter immune function, sometimes with lasting effects. Nutrition profoundly influences immunity. Malnutrition, whether from insufficient calories, protein deficiency, or specific micronutrient deficiencies, impairs immune function and increases infection susceptibility. Protein energy malnutrition causes thymic atrophy, lymphopenia, reduced antibody responses, and impaired cell-mediated immunity. This explains why malnutrition is a major contributor to childhood mortality from infectious diseases in resource-poor settings. Even in developed countries, malnutrition in elderly or hospitalized individuals increases infection risk. Specific micronutrients essential for immune function include vitamins A, C, D, E, B6, B12, and folate, and minerals like zinc, selenium, iron, and copper. Vitamin A deficiency impairs epithelial barrier function and lymphocyte differentiation. Supplementation reduces childhood mortality in deficient populations. Vitamin C supports neutrophil function and is consumed during infections. Vitamin D, beyond its role in calcium metabolism, regulates immune function. Vitamin D receptors are expressed on many immune cells, and vitamin D metabolites influence cytokine production, antimicrobial peptide expression, and T cell differentiation. Vitamin D deficiency, common in populations with limited sun exposure, is associated with increased infection risk, particularly respiratory infections, and possibly with autoimmune diseases. Supplementation may reduce infection risk in deficient individuals, though whether supplementation benefits those with adequate vitamin D remains unclear. The enthusiasm for high dose vitamin D supplementation as an immune booster often exceeds the evidence, a pattern seen with many supplements marketed for immune support. Zinc is required for multiple aspects of immune function. Zinc deficiency impairs thymic development, T cell function, and both innate and adaptive immunity. Elderly individuals and those with chronic diseases often have inadequate zinc status. Zinc supplementation may reduce duration and severity of common colds if started early during illness, though the evidence is mixed. Chronic high dose zinc supplementation can impair immunity and copper absorption, demonstrating that more is not always better. Iron presents a paradox. It is essential for immune cell function, but many pathogens require iron, and the body sequesters iron during infections to limit pathogen growth. Iron deficiency impairs T cell function and phagocyte activity, but iron supplementation during active infection may worsen outcomes by promoting pathogen growth. The appropriate balance differs by context, with iron supplementation beneficial in addressing chronic deficiency but potentially harmful during acute infection. The diet's overall composition, beyond individual nutrients, influences immunity. Mediterranean style diets rich in fruits, vegetables, whole grains, legumes, nuts, olive oil, and fish, with moderate amounts of fermented dairy and limited red meat, are associated with reduced chronic inflammation and lower rates of cardiovascular disease and some cancers. Whether these benefits reflect immune modulation or other mechanisms remains incompletely clear, but such dietary patterns support health broadly. Obesity creates a state of chronic low-grade inflammation. Adipose tissue, particularly visceral fat, secretes inflammatory cytokines, including TNF-alpha and IL-6. This chronic inflammation contributes to insulin resistance, cardiovascular disease, and possibly increased cancer risk. Obesity also impairs specific immune functions, reduced T cell proliferation, impaired antibody responses to vaccination, and increased infection susceptibility. Weight loss reduces inflammatory markers and may improve immune function, though the relationships are complex and not fully understood. Physical activity influences immunity in complex ways. Moderate regular exercise appears beneficial, associated with reduced respiratory infection risk and improved vaccination responses. Exercise increases circulation of immune cells, enhances neutrophil and NK cell activity, and may promote immune surveillance. However, intense prolonged exercise, like marathon running, temporarily suppresses immunity, creating an open window of increased infection susceptibility lasting several hours post-exercise. Elite athletes have elevated rates of upper respiratory infections, likely reflecting both the immunosuppressive effects of extreme training and increased exposure in competitive environments. The mechanisms linking exercise to immunity involve multiple pathways. Exercise induces transient increases in stress hormones like cortisol and catecholamines, mobilizing immune cells and altering their function. Regular exercise reduces chronic inflammation and visceral adiposity. It may influence the microbiome and metabolism in ways that impact immunity. The relationship follows a J-shaped curve. Sedentary individuals and extreme athletes both have impaired immunity relative to those engaging in moderate regular activity. Sleep profoundly affects immunity, though the mechanisms remain incompletely understood. Sleep deprivation impairs immune function, increasing infection susceptibility and reducing vaccination responses. Studies show that one night of sleep restriction reduces NK cell activity and alters cytokine production. Chronic insufficient sleep is associated with increased systemic inflammation. During sleep, prolactin and growth hormone levels increase, while cortisol decreases, potentially influencing immune function. Sleep disruption may alter circadian rhythms that regulate immune cell trafficking and function. The immune system exhibits circadian rhythms, with cell numbers and function varying across the 24-hour cycle. Lymphocyte counts peak during night and early morning, decline during daytime. Shift work and jet lag disrupt these rhythms and are associated with increased infection risk and chronic disease. Whether interventions to support circadian rhythms improve immunity remains an area of investigation, but ensuring adequate, regular sleep appears prudent for immune health. Stress, particularly chronic psychological stress, influences immunity through neuroendocrine pathways. Stress activates the hypothalamic pituitary adrenal axis, increasing cortisol, and the sympathetic nervous system, increasing catecholamines. Acute stress temporarily enhances some immune functions, perhaps reflecting evolutionary preparation for injury and infection risks in threatening situations. But chronic stress suppresses immunity, reducing lymphocyte proliferation and antibody responses, altering cytokine production toward inflammatory profiles, and reactivating latent herpes viruses. Chronic stress is associated with slower wound healing, increased infection susceptibility, and possibly increased cancer risk. Caregivers of individuals with dementia, medical students during exams, and individuals experiencing chronic life stressors show measurable immune impairments. Stress management through relaxation techniques, meditation, or cognitive behavioral therapy may ameliorate these effects, though more research is needed. The mind-body connection in immunity, once dismissed, is now recognized as real and significant. Aging affects immunity inevitably. Immunosenescence encompasses multiple changes: thymic involution, reducing naive T cell production, accumulation of dysfunctional senescent cells, increased inflammation, inflammaging, reduced B cell responses, impaired neutrophil and macrophage function. These changes increase infection susceptibility, reduce vaccine efficacy, elevate cancer risk, and contribute to chronic diseases. Aging is the greatest immunodeficiency, affecting everyone who lives long enough. Strategies to counter immunosenescence are being explored. Thymic rejuvenation through growth factors, keratinocyte growth factor, IL-7, or sex steroid ablation shows promise in animal studies, but remains experimental. Senolytics, drugs that selectively eliminate senescent cells, reduce age-related pathology in mice and are entering human trials. Enhancing autophagy, the cellular recycling process that declines with age, may maintain immune cell function. Caloric restriction and interventions mimicking its effects extend lifespan and health span in model organisms, partly through immune effects, though application to humans remains uncertain. Vaccination remains crucial across the lifespan, but requires age-appropriate strategies. In infants, maternal antibodies can interfere with vaccination, necessitating timing after maternal antibodies wane but before infection risk peaks. In elderly individuals, Reduced vaccine responses may require higher doses or adjuvanted formulations. High dose influenza vaccines and adjuvanted formulations designed specifically for older adults show improved efficacy compared to standard vaccines. Shingles vaccination prevents reactivation of varicella zoster virus, common in the elderly as cellular immunity wanes. The concept of boosting immunity pervades popular culture, with countless products claiming to enhance immune function. Most such claims lack evidence. The immune system already operates near maximum efficiency most of the time. Further boosting could promote autoimmunity or chronic inflammation. What immune function requires is not amplification but balance, appropriate calibration to threats without overreaction or underreaction. Supporting this balance through adequate nutrition, sleep, stress management, and other lifestyle factors is more physiologically sound than seeking to supercharge immunity. Some supplements, marketed for immune support, have limited evidence of benefit. Vitamin C supplementation does not prevent colds in the general population, though it may reduce cold duration slightly in some studies. Echinacea shows inconsistent effects across studies. Probiotics may support immune function by influencing the gut microbiome, but effects vary by strain, dose, and individual. Elderberry, zinc, and various herbal remedies have weak and inconsistent evidence. The placebo effect likely accounts for many perceived benefits, which does not make these interventions useless, just not the immune boosters claimed. Vaccination, by contrast, has robust evidence of benefit and represents the most effective way to enhance immune protection against specific pathogens. Yet vaccine hesitancy threatens population immunity. Addressing hesitancy requires understanding its roots, mistrust of institutions, misinformation amplified by social media, cultural or religious beliefs, and genuine concerns about safety. Simply providing more information often fails. Effective approaches require empathetic communication, addressing specific concerns, and building trust. The COVID-19 pandemic highlighted both the power and fragility of immunity. Vaccines developed in record time prevented millions of deaths, demonstrating scientific capability when resources are mobilized. Yet global vaccine inequity, with wealthy nations hoarding doses while poor nations waited, prolonged the pandemic and allowed emergence of variants. Vaccine hesitancy in some populations undermined herd immunity. Long COVID, persistent symptoms lasting months after acute infection, demonstrates that even non-severe infections can cause lasting harm, possibly through immune dysregulation. The pandemic also revealed how much remains unknown about immunity. Why do some individuals develop severe disease while others remain asymptomatic? What determines the durability of immunity after infection or vaccination? How do variants evade immunity, and can we develop variant-proof vaccines? These questions drove intensive research, advancing our understanding rapidly, but many answers remain incomplete. Environmental exposures influence immunity in ways we are only beginning to understand. Air pollution, particularly fine particulate matter, impairs respiratory immunity and exacerbates asthma and other lung diseases. Chemical exposures, from industrial pollutants to endocrine disrupting compounds, can alter immune development and function. Climate change affects immunity both directly through heat stress and changing pathogen distributions, and indirectly through impacts on nutrition, water quality, and social stability. The modern environment differs radically from that in which human immunity evolved. We encounter novel chemicals, altered microbial exposures, different diets, artificial light disrupting circadian rhythms, chronic psychological stresses, and extended lifespans. Some immune dysfunction in modern populations may reflect mismatches between our evolved immune systems and contemporary environments. Understanding these mismatches could inform interventions to support immunity in modern contexts. Socioeconomic factors profoundly influence immunity and health. Poverty is associated with malnutrition, increased infection exposure, chronic stress, and reduced healthcare access, all impairing immunity. Discrimination and marginalization create chronic stressors with measurable immune effects. Health disparities across racial, ethnic, and socioeconomic lines reflect not just differential healthcare access, but also the accumulated toll of social determinants on immune function and health. Addressing these disparities requires recognizing immunity as embedded in social contexts. Emerging technologies offer potential to modulate immunity in novel ways. CAR T cell therapy, engineering a patient's T cells to recognize tumor antigens, has achieved remarkable success in some blood cancers. Checkpoint inhibitors release brakes on anti-tumor immunity. Tolerogenic therapies aim to induce specific immune tolerance for treating autoimmunity or preventing transplant rejection. mRNA vaccine platforms allow rapid response to emerging pathogens. CRISPR gene editing could potentially correct primary immunodeficiencies. Artificial intelligence analyzes complex immune data, identifying patterns and predicting responses. Yet with these capabilities come ethical questions. Who has access to expensive immunotherapies? How do we balance innovation with safety in fast-tracked vaccine development? What are the long-term consequences of significantly modifying immunity? How do we ensure that immune modulating technologies benefit all populations, not just the wealthy? These questions lack simple answers, but must be addressed as immunology translates into increasingly powerful interventions. The microbiome's influence on immunity suggests that interventions targeting microbial communities could modulate immune function. Fecal microbiota transplantation effectively treats recurrent C. difficile infection and is being explored for other conditions. Probiotics and prebiotics aim to favorably alter the microbiome, though effects are variable and strain specific. Engineered bacteria, designed to produce therapeutic molecules in the gut, represent a future possibility. Understanding and manipulating the microbiome could provide novel approaches to supporting immunity. The immune system's complexity means that no single intervention optimally supports it. Rather, multiple factors in combination determine immune function, genetics establishing baseline capacity, early life exposures shaping development, nutrition providing necessary building blocks, physical activity and sleep supporting maintenance, stress management, preventing dysregulation, vaccination, conferring specific immunity, and medical interventions treating deficiencies or dysfunctions. Supporting immunity requires a holistic approach addressing these multiple dimensions. Individual variation in immunity is substantial. Genetic differences influence susceptibility to infections, autoimmunity, and allergies. Some individuals are naturally more resistant to certain pathogens due to protective genetic variants. Others are more susceptible. Variation in MHC genes ensures population-level immunity, but means individuals differ in which pathogens they can effectively combat. Recognizing this variation is important for both understanding population health and personalizing interventions. The study of centenarians, individuals living past 100, offers insights into successful immune aging. Many centenarians maintain relatively robust immunity, suggesting that extreme longevity involves delayed immunosenescence. Whether this reflects genetic factors, lifestyle factors, or both remains debated. Understanding what allows some individuals to maintain immunity across extended lifespans could inform interventions to support immune aging more broadly. Immunity ultimately serves survival and reproduction, the core drives of evolution. Natural selection has optimized immunity for ancestral environments and challenges, which may differ from modern contexts. Some immune responses that were adaptive ancestrally may be maladaptive now, contributing to autoimmunity or chronic inflammation in environments with reduced pathogen burden. Conversely, modern challenges like novel pathogens, environmental chemicals, and extended lifespans test immunity in ways evolution has not prepared for. Understanding immunity requires appreciating its evolutionary origins and the mismatches between evolved function and modern environments. The immune system is a guardian, protecting us from threats while maintaining tolerance to self and beneficial organisms. It is powerful yet delicate, requiring balance between competing demands. It is ancient yet constantly adapting, shaped by millions of years of evolution while responding to immediate challenges. It is distributed yet coordinated, comprising billions of cells communicating across the body. It is largely autonomous, yet influenced by every aspect of our lives. Understanding immunity reveals both its remarkable capabilities and its limitations. We cannot live without it, as primary immunodeficiencies demonstrate. We suffer when it malfunctions, whether through autoimmunity, allergy, or immunodeficiency. We depend on it continuously, though we are usually unaware of its constant activity, and we are learning to support, modulate, and harness it through interventions ranging from vaccines to immunotherapies. The journey through the immune system, from barriers to cells, from innate to adaptive responses, from memory to evasion to dysfunction, reveals a system of extraordinary complexity operating largely below our awareness. It is a system that evolved not through design, but through the unrelenting pressure of infectious disease selecting for increasingly sophisticated defenses. It is a system that succeeds far more often than it fails, protecting us from countless threats while maintaining the delicate balance required for health. As we rest, as thoughts drift, and consciousness fades, the immune system continues its silent vigil. Neutrophils patrol the bloodstream, ready to respond within minutes to any breach. Macrophages survey tissues, clearing debris and standing watch for invaders. Dendritic cells sample their surroundings, ready to sound the alarm if danger appears. Lymphocytes circulate endlessly, each carrying a unique receptor, waiting for the specific antigen that will trigger their activation. Memory cells, forged in past battles, maintain readiness should old enemies return. This system, this guardian, this invisible shield, asks nothing of our conscious attention. It simply works, day after day, year after year, protecting the integrity of the self in a world teeming with threats. Understanding it deepens appreciation for the complexity of life, the elegance of evolved systems, and the delicate balance health requires. The immune system defends you, quietly and constantly, a faithful guardian from your first breath to your last.